Thank you very much.
Okay. Good morning, everyone. Welcome to our roundtable on leveraging digital innovation to advance One Health. My name is Yannis Kouvekis, and I have the honor of chairing today's discussion. We are fortunate to have an excellent panel today with us. We will start with Professor Sotiris Nikoletseas, who is professor and founding director of the Internet of Things Laboratory, IoT Lab, at the Computer Engineering Informatics Department of Patras University.
Thank you. Zoe, okay, so good morning, everybody. Welcome to Patras. I'm going to talk about the effective blending of two major trends in research and technology, the Internet of Things and AI, into the so-called artificial intelligence of things, IoT, and the challenges and opportunities created by IoT in the very human-centric health domain. As you all might know, the IoT actually represents an integrated system comprised of diverse medical devices, smart wearables, wireless sensors, usually in a home automation environment, and together with a smart clinical infrastructure and equipment, in order to seamlessly collect and process not only physiological but also behavioral data. I guess the very rich diversity of AIoT applications in digital health is based on the, I would say, practically unlimited set of sensors and modalities, such as wearables for heart rate, respiration, sleep, activity recognition, etc., ambient sensors, like for humidity, dust, air quality, also connected medical devices, glucometers, blood pressure monitors, and usually such devices are integrated into a smart home environment, and the massive data they create have to be securely uploaded into the cloud. Some indicative benefits this technology creates for digital health include the remote monitoring of patients and their symptoms, the proactive detection of symptoms and diseases, the reduced hospitalizations, and also the effective handling of chronic care conditions. Additionally, the active involvement of patients into the therapeutic process towards a more personalized intervention. Here is just a very partial list of perhaps visionary but still grounded on ongoing research, examples of applications of this technology in digital health, nanoscale robots, navigating the bloodstream in order to precisely deliver drugs wherever they are needed, smart ingestible sensors to wireless report physiological data, smart ophthalmic lenses, but also personal neurotherapeutics that actually via AI can dynamically adapt to patient's status, like the mood, the cognitive load, and accordingly stimulate several parameters of the patient. Another potential application related to public health is the possible deployment, massive deployment of swarms of wearables throughout a region in order to anonymously collect sensor data from everywhere. But also in the mental health domain, AI can support emotion-aware digital systems. Such unprecedented, I would say, opportunities necessitate addressing critical complementary challenges. First of all, related to data. Data is quite diverse. It can be noisy, can be unreliable, or even missing. Also, the physiological monitoring of people creates a lot of sensitive personal data. So the security and privacy of such data needs to be properly safeguarded. From a computational perspective, several times the health applications are very time critical. So the latency needs to be very low. At the same time, the computational environment, the sensors, etc., they are very limited in terms of resources they can support, the memory, the CPU, etc. So we really have to very effectively manage those limited resources in order to provide real-time processing at the edge of the network. Another critical issue is the usual lack of properly annotated data sets. So usually it's very hard to get a data set which is correctly annotated by expert clinicians and the involvement of properly cooperating patients. Now, for the last two bullets, the human factor gets into play here. For doctors, for clinicians to trust the AI systems, the AI systems have to overcome their usual black box behavior. They have to be transparent, explainable. They have to justify their predictions. So this is called explainability of AI inference. And also, regarding the user adherence to protocols, even when the AI systems are very non-invasive and user-friendly, the cooperation of patients cannot be guaranteed. So the AI systems have to be by design resilient to such unpredictable behavior of users. Now, in the framework of the above, let's say, principles, I will summarize a few characteristic use cases developed in our IoT lab here at the University of Patras. So the first one is about respiratory diseases in particular and how to diagnose them using, on the basis of the voice. This is one line of research. Then how to detect allergies via gesture recognition and also via employing the crowdsourcing paradigm. A third topic relates to investigating the impact of indoor air pollution to health. And a recent research line we're pursuing in my lab relates to intelligent image analysis, particularly for the eye. Let me now go over each one of those elements. So the first one is a direct project we have with Pfizer, the major pharmaceutical company. So we focus on respiratory medical conditions. We have developed both a smartphone application, but also a web application in order to really monitor continuously and detect a few typical respiratory diseases like cough or symptoms like cough, lung symptoms, and asthma, COVID-19, COPD. It's worth noting that we only use the smartphone, so no external device is needed. And also, all AI is run locally on the smartphone. No exchange of data from the smartphone to the cloud. So actually, our architecture is a bit, let's say, hierarchical. So first of all, we have this passive cough analyzer, which runs in the background of the smartphone operation, and if needed, then the patient is undergoing an auscultation by placing the smartphone on their chest, so the smartphone acts like a smart stethoscope. Another important point is that, as said, it's always hard to get enough good data. So, and maybe this is of more general interest, we have effective methods based on AI on how to create synthetic data, which, however, should be very similar to the actual data, the real data by patients. So this is called synthetic data generation, and the methods we use are based on generative AI. Obviously, we had to implement quite sophisticated digital signal processing methods. I will very briefly highlight a few ones. Here you see the frequency spectrum. We truncate the spectrogram by focusing only on the more relevant part of the frequency spectrum. Here we apply some low pass filter, high pass filtering in order to remove noise. And finally here, we use some spectrum calibration methods in order to boost the more informative parts of the spectrum. I'm glad to report quite satisfactory results. So in all cases, we came up with a precision accuracy above 86%, even up to 93%. The performance is quite good, so it takes only 5 seconds to analyze cough in real time, only 1 second to analyze the lung sounds during auscultation, and the footprint of the software we develop is quite small, so it can actually be uploaded and operating into even, let's say, less sophisticated smartphones. Now, beyond the automated analysis, we also glad to provide doctors with assistance. So here is like a dashboard for the cough analysis. So here you can see the number of detected coughs, the duration of the cough, and eventually the classification of the different cough types into the various diseases, normal, asthma, and COVID. Also, for the auscultation part, the lung sound analysis, again, we provide historical data, but also the classification of the lung sounds into the various medical condition classes. Okay, let me move to the second research line we have implemented. So here we try to detect allergy via a complementary, I would say holistic approach, combining objective information coming from, let's say, open source platforms or fixed stations, together with more subjective perception of allergy from wearables and also using crowdsourcing. So this gives rise to the paradigm of hybrid human-machine knowledge. Now, for the first part, let's say the subjective part, we model the allergy detection problem as a human gesture recognition problem. So actually, this is based on a hypothesis formulated by the medical experts in our project, who hypothesized that at the onset of an upcoming, let's say, allergic situation, the patients tend to make some typical gestures, rubbing their nose, ear, etc. This is only a selected list of typical gestures, and in order for us to train the AI systems, we need to really collect data, both from a hospital in Athens, real patients, but also in the laboratory from volunteers. So we provide them with a wearable, okay? And we collect the data collected by the wearable. Indeed, we can see here that the AI system is really able to classify, to identify distinct patterns for different gestures. So this one here corresponds to the palm rubbing the nose, while this figure here represents the case where a patient vibrates their finger in the ear channel. I already referred to the data challenge. So here we provided, perhaps again this is of general interest, a very user-friendly application which directly collects data from the wearable to the browser without necessitating any additional devices. And this is the time series created by a patient wearing that smart wearable. And indeed, the identification of the gestures is quite precise. Here you can see the raw time series sequence of a person writing on their laptop and at some stage making some gesture, okay? And you can see here on the right that indeed our system is able to very precisely locate, identify that gesture and separate it from noise. An important extension of this system is the so-called allergy map tool. So here, people register in the platform, and the idea is that from the user, we get subjective perception of allergic symptoms in a VAS tool. So it's a visual analog scale system where basically the patients grade their subjective feeling in a scale, let's say from 1 to 5. I'm good, I'm very good, or very bad, or whatever. And this comes together with spatio-temporal recording of the place where this feeling is obtained. And then this is combined with objective information, localized measurements related to irritants, humidity, pollen, dust, collected by fixed stations, meteorological or other, and open source platforms. So here on the left, you can see the Patras area, and this is humidity monitoring in the Patras area. This is dust monitoring, African dust in particular, and here you can see a map of allergens in the broader Patras area. Now, a next step we undertook is the following, that we introduced social media analytics, and particularly Twitter posts, in the sense that we monitor in Twitter posts related to, particularly in this case, allergic rhinitis. And the idea is that when there is like an allergy outbreak, an upcoming allergy outbreak, the frequency of relevant posts, Twitter posts, gets higher. So basically, this represents a complementary holistic platform. From fixed stations and platforms, it gets pollen, dust, and humidity information. From social media analytics, it focuses on the frequency of relevant tweets, and subjective perception is collected via the VAS tool and via smart wearables the patients are carrying with them. And actually, we implemented this approach in the US region, and we came up with this hybrid spatio-temporal mapping of the exacerbation of rhinitis in USA. So actually, this gives rise, I would say, to a new complementary sensing paradigm incorporating human perspective, sensing technology, and also machine intelligence. Now, let me now move to a third activity. It's about the impact of indoor air pollution to health. This was particularly relevant in the COVID area, but it's always relevant. So again here, this research project researches a medical hypothesis that the different pollutants, chemical, biological, whatever, may exhibit synergistic effects when it comes to their impact to pollution. Superadditive effects. So the simultaneous presence of different pollutants may have impact which is much bigger than just the sum of the impacts of the individual pollutants. And this is tested and validated in several schools in 5 countries around Europe. And this is a preliminary evidence that shows the correlation between COVID and pollen. So basically an empirical explanation of this is the fact that the pollen molecule, which is quite big, can carry with it the COVID virus. The main questions we investigated are the following. You have an indoor place. How pollution disperses in this, let's say, room or classroom or whatever? This is very critical in order to be able to estimate the personalized exposure of each one of the students in the classroom. I already referred to the synergistic effects we want to investigate. The explainability and causality. So causality is different than correlation. Causality means that a certain pollutant has really a causal effect to a certain health outcome. And finally, we also implement some mitigation actions, some novel air purifiers, but also natural green walls. Now, let me focus a little bit on the dispersion problem. So here we have a cooperation with well-known chemical engineers. They use the so-called CFD mathematics, so it's computational fluid dynamics, on how a pollutant spreads in an indoor space. This is very precise, but it's very abstract because it assumes a very idealized setting, no furnitures, etc. It's very abstract, and it's very heavy computational. So my team, we cooperate with them in order to complement their very detailed but very computationally heavy approach with a data-driven method. Okay? So basically, we deploy some sensors, a few sensors. We collect the actual measurements, and then we implement the so-called virtual sensing or soft sensing method, which basically is the following, that based on the real measurements in a few places, our algorithms can extrapolate the pollution levels in very different places of the room. So actually this helps, this complements the CFD approach by keeping its prediction power quite high, but at the same time by lowering its computational load. As far as investigating synergies and causality, first of all, we create a very diverse, heterogeneous data lake where immense volumes of very diverse data is flowing in, data from sensors, from actuators, from wearables, questionnaires, gamified apps played by the children in the school. And of course, we use state-of-the-art neural networking and other causal machine learning methods in order to be able to infer causality and superadditive interactions. Now, another quite recent activity in my lab relates to image analysis, particularly for the eye. So the idea is that first, because of privacy concerns, we have to do the so-called sclera segmentation. So you know that in the near future, we will be able to unlock our computer with our eye bank account, so it's very critical. So before doing anything, we really have to remove the eye information and only keep the sclera, the white part of the eye, and this is called sclera segmentation, and it is done by a suitable AI system. Okay, so we do the eye sclera extraction. So this is the eye. This is the eye after removing the critical information, and the white part is the sclera. And we also created a web tool for doctors to be able to annotate what they see on the white part of the eye, and to also insert into the system clinical data about the patient, blood examination results, X-rays or whatever. And we surveyed different public data sets and created a database of more than 500 eye images. And the idea is to really investigate possible correlations between the yellowness of the eye with important biomarkers. So this is a preliminary finding that indeed the yellowness of the white part of the eye can be very informative for a very rich variety of conditions. Okay, so far I focused on digital health for people, but this conference is about the One Health concept. So in our lab, we also deal with animal health, how to monitor the vital signs, the stress, particularly of sheep, how to real-time monitor barns, temperature, humidity, air quality, etc., and particularly how to detect mastitis, and overall, how to use AI in order to come up with early warnings for diseases, discomfort, and low feed intake. How I'm doing on time? Hello? How much time do I have? I need 3 more minutes. Is it okay? Okay. Okay, so a final research line we are investigating is the following. How to protect birds from colliding with wind turbines. This is really very important because the expansion of deployments of wind turbines everywhere. Now, when it comes to agriculture, we investigate water scarcity, water quality, also crop monitoring, particularly at the microclimate level, soil, humidity, radiation, etc., and also how to predict irrigation and plant stress prevention. So actually, and this is the vision of the conference, we would like to really come up with a continuum, digital continuum for One Health, particularly in the era of climate change, which really affects a lot the One Health aspects. And the vision would be to eventually come up with a unified digital ecosystem for human, people, plants, birds, everything, via seamlessly and continuously collecting diverse, massive data from everywhere, animals, people, agriculture, the environment as a whole. Thank you.
So, are there any comments or questions from the floor? Yes.
Okay, thank you very much for your interesting talk. I just wanted to know about the movement recognition the movement recognition in case of asthma.
Which recognition?
Movement recognition for asthma.
What do you mean by movement recognition?
You know,
the gesture recognition.
Gesture recognition. Yes.
Yes.
Is it somehow linked to countries or geographical localization or ethnical origin of the persons, or can you just include any people to this program?
Yeah, let me tell you. So actually, you are referring to that part where we deliver the wearables and we monitor the gestures of the people, right? I would say the following, that this tends to be rather universal, but the complementary aspects like the exposure, let's say, to pollen or to dust, probably, I mean, obviously, really depend on the peculiarities of the particular country. So in the south, there is more dust, less dust in the north, etc. So I would say the gesture recognition itself doesn't seem to have a strong correlation to geography or to countries, probably, as far as I can tell. I'm not a medical expert. I'm doing the computational part.
So you've done only this research in Greece, or did you have interclinical
No, this one was done in Greece, but the one about the crowd sensing eventually investigated the USA.
Okay, thank you very much.
Thank you.
Are any questions from no? Okay, I have one question.
Please.
At what extent, at what level the explainable artificial intelligence being implemented in such systems as like the wearables devices, etc.?
Well, as you know, because you are technical too, right?
Yes.
It's always challenging. I think that in some cases we have done quite satisfactory progress, so we can really identify the factors that dominate the outcome of the neural network or whatever. But it's always challenging. Particularly causality is very challenging, and it's rather new. I believe that in the state of the art, causal ML is not investigated as it should be so far. I believe it's an open question and topic.
Yes.
But we have done some progress, yeah.
Thank you very much.
Thank you.
Yes. Just one moment for the microphone.
Congratulations for your presentation. I wish to ask something about the animals. What do you measure in order to get some data from the animals, from the sheep with mastitis, clinical signs of disease, and what about the milk, the deviation of the parameters?
Actually, we put some wearables on the animals.
Okay.
And we measure several vital signs of them, and also in the system that, I don't know the exact term, which actually collects the milk, even in that system we deploy some measurement device. So we combine information from the animals themselves and also from the milk collection process.
I'm interested with that. We will discuss it later.
Sure.
Thank you so much.
Thank you.
Thank you very much.
Thank you.
Let's continue with the second speaker, Professor, hope to say correct, Mario Munoz Organero from he's associate professor in the Department of Telematics Engineering in Spain, and he will speak about using machine learning models to anticipate the spread of diseases.
Okay, let's share the screen. Okay, perfect. So thank you very much for the invitation and for having me here. And the topic of my talk will be using machine learning models in order to use the data that we have about the spread of a particular disease, in this case is respiratory viruses. So using machine learning in order to be able to predict, to anticipate how the virus will be spread. So this is the topic of the talk. My name is Mario Muñoz, and I work in the University Carlos III in Madrid, and I lead the research group in machine learning, especially applied for health health applications. Okay, so the problem that we want to solve is estimating the spread of a respiratory virus. We have different approaches that we can follow. For example, one first approach could be using mathematical models. One can try to model the biological process inside the human being, and using mathematical equations, we can try to have a model that more or less behaves similar to the human body, and we can study how the virus spreads inside the human body and how the virus spreads over the air, and so on. Here you have an example of one publication and some equations taken from the publication, in which we can see, for example, some differential equations trying to model how the virus is produced inside the infected cells. Well, this is one approach that we can follow. We can try to have a good mathematical model that tries to take into account all the possible variants affect the spread of the virus, and solving these equations will give us an estimation about how the virus will spread over time and space. Another approach that we can take is using simulators. In that case, we can have a software agent to model each individual in a population, and we can have these software agents modeling people with belonging to some communities, and we can apply some rules to these agents, some mobility rules, whether these simulated agents are following lockdowns or are vaccinated, or all of these policies can be introduced into the software agent, and then we run the simulation in which we allow the agents to interact, and therefore we can try to mimic what people are doing and try to somehow estimate the spread of the virus. And the final approach, that is the one that we are going to take in this talk, is using machine learning models. Machine learning models are again models, but not necessarily related to the biological processes behind, are more or less generic model with some weights that can observe data and adjust the weight so that they can be fit to generate the output that we want from them. In our case, we can fit the models with the data that we have related to the spread of the virus, and we will let the model to try to estimate how the virus will be spread over time and space. Of course, these three families of approaches are not incompatible. Just to mention an example, here you have one of our publications in which we combined these three types of model into a meta model to try to achieve better predictions. And it was applied to the spread of the virus in the Madrid region. And you can see that we have applied three different models. The first one is our machine learning based model, but we have another mathematical, and we have the epigraph is a simulator. Each of these models was able to produce predictions, which you can see here compared to the real one. We try to estimate the incidence of the virus. In that case, COVID is the major example in these slides, not because COVID is the most important, but because we have more data related to COVID, so it's easier for us to apply machine learning models having more data and achieving better results. So in that case, you can see that each of these different approaches is trying to properly predict how the virus will be spread, but of course, all of them are not perfect. And in that study, we try to create a meta model that combines the three of them and produces a better outcome. In this case, you can see that the errors that we have in each of the models is higher than the ensemble approach, the combining the three models together. Okay, but for the talk today, let's concentrate on machine learning models. So this is the scenario we have. We have data, and we want to learn from the data, the different patterns that will allow us, that having the data will allow us to predict what is coming next. So what type of data can we use? When we want to try to predict the spread of a respiratory virus. So we can have incidence data, which is the time series of the number of infected people over time. This is something which is quite important because it's the basis of what we want to estimate in the future. We also may have information about how this incidence is spread over space. So we may have maps of incidence data. Then we also know some calendar events. We can know if we are during the weekend, we can be on holidays or so on. So this also is important because it affects mobility, it affects other variables that can have an impact on the spread of the virus. Then we have mobility data. Mobility data is also important when trying to anticipate how the virus will spread. And for mobility data, we will have to use some proxy variables. Traffic is a proxy variable, but we are going to see two examples in the slides today, how two proxy variables can estimate the mobility of people. Then we have meteorological data, and we also have data related to policies, such as, for example, vaccination policies, using masks or lockdowns. So all of this information somehow is related to the spread of the virus. So on top of this data, we are going to put a machine learning model. So we are going to put a model with some parameters, and using previous data, we are going to optimize the values for the parameters in the model so that the predictions that we get are as optimal as possible for new data when trying to do some inference for new data. Okay, so this is more or less the framework: having some data, having a model, letting the model learn the patterns in the data, and then using the model for new predictions. Okay, so let's start with simple models that only use the time series that come from the incidence data. So let's assume that we only want, we only have this incidence time series, and we want, based on previous data, we want to make the prediction about how the spread of the virus will come next. So we can use different models that are different machine learning models that are designed for predicting in time series. Maybe the most basic one is the autoregressive model. In this model, we can see that using this are the variables of the time series in the past instance of time, and this is the value that we want to predict. So we will have some weights that can be learned. Using the past data in the data series, we want to learn the optimal parameters so that when we put the model to do some inference with new data, this will generate the best estimate for the coming values of the incidence data. So this is a basic model, but the idea is that in any machine learning model, we will have some data based on the past values. We will have some weights that we have to train. And when the model is trained, we can try to use the model for doing predictions. Okay, this autoregressive model has been, of course, improved by other versions like RIMA or SARIMA. And well, this is one family of machine learning models that we can use. We have based our research mostly based on recurrent neural network, which is another family of models that we can use that are a bit more complex than the autoregressive model. In that case, again, we have the time series information. So for each instant of time, we will have some data. It can be a single value or it can be multiple value if we have several time series. Let's say that we are measuring the incidence data in several points in space. We can fit this data into a multivariate time series. Okay, so each instant of time will be fed into the model, but now the recurrent neural network, it will not be just a simple weight applied to the data set or to the data point. Now we are going to have something a bit more complex in which we are going to combine some neurons inside with some weights. So for each instant of time, the processing of the input data will be a bit more complex. That this will give us more flexibility or more power to learn more sophisticated patterns in the data. So putting something like that with several neurons, this sigmoid here represents a neuron like this. So putting this model in each instant of time, feeding the model with the current data point, and as you see, we have an arrow that fits the past predictions to the next time step. So we have everything connected, so we are able to do prediction based on all past information for new data. Okay, there are many types of models that we can use, temporal transformer, many types of models that we can use. In the slides, we are going to present some of the results that we have obtained. And of course, we can use to do predictions on different time series. Here you have the incidence, here you have the cumulative cases, or you can apply to deaths or whatever. Okay, we can add more information to the machine learning model. The idea is that the more information you add, the more power that you have in your hands, so that you expect to have better predictions. So now let's say that we want to add a spatial information to the model. We have generated well, this is a map of the Madrid region, and the map you have the incidence value for COVID in a particular instant of time, a particular week. So you have in brighter colors, you have higher values of incidence. So this is a map representing for an instant of time the spread of the virus in the Madrid area. We can have a sequence of images representing the evolution of the spread of the virus over time. And now we need a model that is able to extract patterns from this information in order to generate what? To generate how the map will be next week or in 3 weeks. So this is what we want the model to generate. An image that captures how the virus will be spread on a future instant of time. Now we need different machine learning models able to extract information from images, and typically models are based on convolutional neural networks. Again, every machine learning model is a set of weights that have to be trained on the information. Now our weights will be filtered like this. Each box in the filter is a weight that will be put in a region of the image, and the filter will move over the image to generate a new image. So the idea is that we are going to have to train these weights so that the result that we get is the optimal one. The optimal one is having a map which is as close as possible to the real one. In our case, we have 2 dimensions. We have the spatial dimension with incidence map, but we also have the temporal dimension because we have a sequence of images. So what we did in this, well, you have the reference of the publication. What we did in this study is using first a convolutional image to extract information from each image, then summarizing the results into a multivariate output, and then feeding this multivariate output that represents the information in each image to an LSTM. The LSTM will extract the patterns over the time. So we wanted at the end to generate the final incidence image, and well, you have here for a particular case 2 predictions for 2 points in the image, in which you can see more or less that the predictions follow the real values. And well, you have some comparisons that you may find in the publications about the results of using simple temporal analysis models, simple spatial models, or combining both models in a single, both temporal and spatial models into a single one. And you can see that in our case, we are able to achieve nearly optimal results, or results that are comparable to other results in other areas using similar models. Okay. We can make the models more complex. The more data you have, you expect to have a better result. So an important variable that we think that well, it's been shown that it has an impact on the spread of the virus is the mobility of people. We don't have the capacity to know the exact mobility of people, but we can use proxy variables to estimate this mobility. So a first proxy variable that we use is the traffic data. We have over 4,000 traffic sensors in the city of Madrid. Here you have each point represents a traffic sensor, and more or less this is covering the entire area of the Madrid city. And we have divided the sensors into two types. On the one hand, highway sensors and city sensors. We didn't use the highway sensor because highways are more not quite related to people moving inside the city, but people passing by, and doesn't necessarily mean that are people that will interact with the city, with the inhabitants in the city. But using the urban sensors, the sensors in the normal streets of the city, you may have an estimation about how much the people in each area is moving. It's a proxy variable, so of course it's not perfect, but it's something that we could try to use to try to better optimize the results of the models. Well, in our case, we have the capacity to process images, so we have converted traffic into images. So you have here an image that more or less captures the intensity of the traffic in each part of the city. On the other hand, we have the information about the spread, the incidence of the COVID virus in this case, and we have the different primary care centers where they conducted the tests. So we can also create images about the incidence of the COVID virus in the city of Madrid using the same resolution as the traffic image. So we can have data related to mobility and data related to the spread of the virus. So once we have these images created, of course, we can generate an image per week. So we have a time series of images. Then we can try to develop a model, a machine learning model that is able to try to find the patterns in these images in order to try to generate how the virus will spread in the next week, in the next 2 weeks, and so on. So we developed two different models. The first one, combining both images into a multicolor image. You have here to have these two means that these two images are combined together into a single image which has two colors, the traffic and the incidents. Or we have developed a similar model, but not combining the images, but processing each image, the traffic image and the incidence image independently. And of course, at the end, you combine both at the LSTM part of the model. So both models are trained so that they are able to, or they try to predict how the incidence image will be in the future. And here you can see the results. We tried to see, is traffic enough to predict how the virus will spread? And you can see that not too much. The first image that you have on the screen is only using traffic data, nothing else. Can we predict how the virus will spread? And you can see that the results are not very good. So for example, in this case, you predict that the virus will spread while the virus is decreasing, so that the traffic itself, it's not enough to predict how the virus it will develop in the future. We also try to apply a model only having this is what we saw before, only having COVID data. We had better estimations about how the virus will spread, but what if we combine both? What if we combine traffic and COVID data? And we have models that better predict, so you can see for the independent images or for the combined images, models processing both information together, and you can see that in both cases we can have better estimations about how the virus will evolve in the future.
Sorry,
okay,
sorry,
so
Professor,
one,
we have
yeah.
5 minutes.
Okay. So I will try to condense the information. Thanks a lot. Okay. So, well, traffic was a proxy variable that is somehow able to predict or correlate with mobility, but we found a better variable to estimate mobility of people, and this was the use of the bike service, the public bike service in the city of Madrid. Here you have the city of Madrid divided in districts, And on the left, you have the location of the health centers for the COVID images. And on the right, you have the location of the bike stations. So the city of Madrid records each trip that each user does using the bikes. So you have the information about all the trips per day between each pair of stations in the city of Madrid. So this is a better proxy of how people is moving from one district to the other district. Of course, not all the people moving from one district to the other using the bikes, but the bike could be a better proxy that using traffic data, which is more, well, it's less related to movement from one place to another. Okay, so using this data, well, we try to, again, to create a model, to feed this information into the model, and we found several interesting results. Not only that we got good results in predicting the virus, but we also saw that we got better results for parts of the city in which we had a higher amount of bike stations. You can see that you have an inverse correlation. The more bike stations you have, the better results that you get. So the errors are small. So this is something that the more, of course, if you want to have a better estimation of mobility, if you have more bike stations, you get a better idea. Okay, more information that we can add to the model, and now it comes the weather. Weather data is also quite important for predicting the spread of the virus. So we also developed some models based on some data that we also have for the city of Madrid. We have the temperature, the precipitation, solar radiation. So we put all of these time series, all of this information into the model. Well, and the predictions here, you have the R values that should be close to the diagonal. So you can see that we had some error, but more or less the predictions were quite good. And to finalize my presentation, I have also captured two more publications from other people doing similar results, similar studies. Here you have another publication about using weather information for predicting the number of hospitalizations of people needing help related to respiratory diseases. And well, this study was interesting because they also, among all the variables that they took into account related to weather, they detailed a study about which variables were more important when getting the results. But well, since we don't have a lot of time, I will not go into the details of this study. And related to animals and other use of machine learning models, can we use machine learning models for estimating movement patterns or the spread of other animal-related diseases? And in this case, we are starting a project related to predicting how the olive fruit fly plague will evolve based again on machine learning model. We still don't have data for that, but I have put just an example. Other people doing similar results, estimating how this plague is evolving based on satellite images, and of course, and meteorological data, and of course, you need some data for the real target values, and in this case, they are using traps in order to estimate how the real data is doing. Okay, so thanks a lot, and if you have any questions, just happy to answer.
Thank you very much. Are any questions from the floor? Yes, one here.
Hey, thanks for a great talk. So to run these machine learning algorithms, do we need high incidences in general, or is it possible also for very low incidence virus and viral infections?
Well, I think what is important is to have as much input data as possible. It doesn't, I mean, it doesn't necessarily correlate it with the high values of incidence, but having enough input data. So it doesn't matter if the incidence is small, but as long as you have a long time series of data, you can try to fit the model and do the predictions.
Okay. Okay, I have a question. Have you used machine learning algorithms or models in order to find more appropriate deterministic models? Because in the first slide,
Yes.
you present the differential question model of virus disease. So have you used ML in order to find?
Yes.
Yes.
Yes. We have also used machine learning to optimize the parameters in mathematical models. So at the end, in mathematical models, you have to adjust them to particular individuals and so on. So you also have some parameters. So for estimating that parameters, you can use machine learning models. Yes.
Very interesting. Thank you very much.
We have no other questions.
Okay. So thank you again for your
Okay.
presentation.
Thanks a lot for inviting me. Very happy to be here with you. Thanks.
And we will close with the third speaker, Evangelia Zani. I think this is the name, okay. Who is associate professor and head of the Department of Epidemiology in School of Medicine in University of Ioannina. She's online.
Good morning, everyone. I hope you can see me and hear me well. Is this the case?
Excellent.
Wonderful. So, good morning. I would like to thank the organizing committee for the invitation. I would like to congratulate the project for the work done so far, and I would also like to compliment the previous speakers for such an elaborate work and very relevant to the work we all do on the field. So my name is Evi Zani. I am the professor of epidemiology and head of the department here at the University of Ioannina at the School of Medicine. And I also serve as an adjunct professor at the university, at Brown University in the US, at the Department of Health Services Policy and Practice. So my talk will be a little bit about the evidence synthesis, which lies in the core of my work across health. And today we will discuss a little bit more about the challenges, the limitations, and the possible solutions related to One Health data ecosystem. So there will be a rather general part describing the challenges and the second part where we discuss our work and the solutions. So, of course, this is always the case. I know you're quite familiar with this slide. This is the general ecosystem of One Health. What I would like to remind us all is that we pace within a structural institutional framework, and this is not the case for all new domains. One Health quite very swiftly got into the institutional realm, which is not the case with other domains, and this creates challenges within the field. So as any new field, any novel field, we know that there is knowledge generation, data generation, which we want to transform into knowledge. And here indeed, we can see that the rate of publications that are related to One Health and the related information that is gathering is, of course, accumulating over time. What I would like to point out here is that this is only the tip of the iceberg. We have a very, very large number of additional data sets which are handled and processed internally, which are directly relevant to public health and which are in One Health and which are not visible within the scientific community automatically. When we try to map the ontology of the field, we see indeed that we see the usual suspects: drug resistance, antibacterial agents, global health, surveillance, gender-specific differences, the common pathogens. But we know now that One Health is far more diverse than what we just see or what we in the beginning envisioned. And indeed, we have here the evolution of also to say of this knowledge transfer, of this idea of going through the various aspects of the One Health framework itself, and we have the theory of change. Because we know that for One Health to be implemented, indeed, a change needs to be implemented. The only thing I would like to pinpoint is the density of this slide. So you can see that we start, for example, with anthropogenic influences of health, animal health, human health, environment health. And then we move, and this is the place that I will focus more, on the third pathway, which is related to data and knowledge generation. So indeed, the integration of data and knowledge generation within each platform is a challenge on its own. Are there any data available? We know this is a philosophical, a rhetorical question. We have a very large amount of data that are somehow at some point available in some form. We have distributed data, we know that. We have made progress in aggregated data, and we know that we have a lot to do related to integrated data, which is the optimal goal for One Health. The One Health surveillance system workflow also has specific challenges, usually across different things. I originally come from genetic epidemiology. We had the more or less the same matrices, and we had problems with the analysis, harmonization, QC of the data, etc. Here we have multiple settings, multiple matrices, multiple analysis methods, multiple analytical methods, and that creates the huge heterogeneity of data that I will discuss further on. And of course, surveillance systems, as I said, are institutional by their nature. So they have to be embedded into a system. Here is a typical system development where we start with the scope and we end up with the implementation roadmap. And here, knowledge transfer is important and evidence synthesis. Moreover, challenges arise from transdisciplinarity, which also poses the strength of the field. We know that it is not only about interdisciplinarity, where many people sit around the same table, but it is also transdisciplinarity, which means that we have to endorse attributes of different disciplines and become something different from what we started being when we began working on One Health. And finally, vulnerable populations. There is no other domain, scientific domain, where vulnerable populations are so much incorporated into the core aspects of this new, so to say, new field, not only scientific, but policy field as well. And of course, I don't have to remind you, it was also pinpointed by the previous speakers that the mode of action of the associations and the causal relationships that we are discussing are related to a vast majority of exposures that is related to the environment, the humans, and animals. So we have the personal exposome, the external exposome, and the biological responses that are related to this exposome, which is again something of very specific approach and of needs that are quite important. A final thought before discussing and summarizing the challenges is that we have the one sample many analysis already in place, and it will be in full swing later on. So here again, we have to see the ethical challenges behind that for data sharing and data harmonization and all the other remaining challenges. And just to give you an idea of the varsity and of the veracity and versatility of the stakeholder matrix, these are all the systems colleagues before discussed how their own disciplines come into that new era, into that new realm, and how socioeconomic fingerprints, environmental fingerprints can collide together with statistics and modeling to offer and produce information systems that are quite into the digital connectivity up to the commercialization of the field itself, which is also, as you know, very active, not to say aggressive. Different fields, this is part of the work we do on surveillance with other colleagues across the pond. So here we can see that different surveillance systems can take different attributes, whether somebody it's a personal data, like a vaccine, or a minority status, or epidemiological data that come in on block, etc. And finally, to connect with a colleague that gave his lecture first today, it is this huge challenge of what people in the field already call One Digital Health. It is the merging of One Health and digital health, where the various ecosystems can merge, can integrate, and where technology takes precedence, as we already know. So to summarize that first part, what is the status of the evidence? I would like to highlight that we have the vast majority of the evidence is fragmented and heterogeneous. There is a lack of standardized terminologies and ontologies. Data accessibility and governance barriers are already in place and are still there. There is inconsistent data quality and completeness across the different sources, difference in methodologies across disciplines, limited integration of qualitative, quantitative, and genomic data, and of course, the lack of computational and analytical infrastructure that was also indicated in the previous lectures. The difficulty in identifying causal pathways, us as epidemiologists are very sensitive to that particular attribute. Challenges in timeliness and real-time synthesis, institutional and cultural barriers, and last but not least, limited capacity and training. One Health has been very sensitive in identifying capacity building avenues and also training opportunities for all types of researchers. So there are a lot of international initiatives. Why? Because we, within the One Health realm, we have our eyes set also at the developmental goals. And we know that without appropriate data and implementation of evidence-based policies, achieving these goals will not be manageable. So having said all these, I will pursue into discussing with you some of the solutions that we have about the evidence challenge and the evidence integration based on the evidentiary needs that are there. So our solution and what we do, we have already 4 running projects on that, are the evidence maps. Evidence maps are an evidence synthesis tool that allows decision makers to rapidly assess a body of evidence relevant to their policy questions. So evidence maps are related to policy making. Based on a rigorous and transparent research methodology, they collect, organize, and appraise different forms of policy-relevant evidence. And this evidence can then be mapped into a policy framework and have an interface that is interactive where it can be visualized. So where can evidence maps be useful with? They can be used as a scoping tool. This is the most usual, so to say, function of them. When they set policy objectives, because they are, in the beginning, they are considered scoping review variations. They can also be used as decision-making tools, as engagement tools, as organizational tools, but most importantly, they can be considered as knowledge management tools where people can function within a repository of easily accessible and policy-relevant evidence. A research tool also is a function of an evidence map where it can identify gaps, coverage, and patterns in the available evidence on the policy question. And it also is an accountability tool, which is also for internal value, but can be used across different organizations. And of course, for high-level policy making, a research commissioning tool is the standard use of the evidence map. So evidence maps are quite flexible and adjustable. They can use different questions for different lines of evidence. So we can incorporate qualitative research, quantitative research, audits, epidemiological surveys, epidemiological data, and as long as all these lines of evidence have a way to be incorporated into the map. So in the construction pattern on the map, we want this aspect to be incorporated. And of course, we know now today that when we interact as scientists with the policymakers, the way of functioning has changed. We have the push efforts, which are our usual, tactic up until the previous years, where researchers would reach out to policymakers to show what the research agenda is about, etc. We have user pull efforts when the policymakers realize that the need of an evidentiary basis for policymaking, they reach out now to us asking for more information. We have exchange efforts where we come together, and we have an intermediate where we have integrate efforts where researchers and policymakers interact through these knowledge translation platforms, such as are the evidence maps. And of course, the evidence creation funnel creates different levels of certainty around the accumulated evidence. We have experimental studies, observational studies, qualitative studies, as I mentioned, and then we proceed in the synthesis in order to be able to produce products. For example, in One Health, we don't have HTA activity, very active health yet, but we have policy briefs, a lot of them. So what does an evidence map look like? An evidence map looks like that. This is the general structure where you have an outcome. This can be a systems outcome, or it can be an intervention, or something that is related to what we discuss. Then again, it can be a map that relates to the provenance and the characteristics of the evidence. It is an important thing to say that basic science has a distinct position in the evidence map, as also other parts here. These are examples of additional and experiment of other efforts across different fields. Also, we could have interventions because we know that within One Health, there is the possibility to have system-wise intervention, as I mentioned before. And of course, we can have population-based evidence maps. What is important to understand is that we can have uncertainty maps. So here, this is an effort that we have done on air quality and health-related outcomes, where we have seen that people also try to understand and identify causal relationships within an area without taking into consideration the robustness of the evidence. So a weight of evidence map is a way of understanding how the specific associations and when are related to certainty that is related to the validity, the internal validity of the associations and the studies that support that specific association. Just to give you an idea, here we have certain air quality attributes, and here we have the levels of evidence. We have not significant associations, weak evidence, weak grade, we have suggestive evidence, highly suggestive and strong. And you see every bubble here, every small dot represent a specific synthesis effort within the air quality area. So these are all, to say plainly, all meta-analysis. And what we see, when we accumulate, when we synthesize all this evidence, we end up with only a few disease entities that are robustly associated with air quality attributes. And finally, another thing that is important is the evidence gap map. The evidence gap map is an idea where people can understand within the specific research agenda where there is the need for further policy focus and policy emphasis related to further research. Here, for example, is an evidence map that is commissioned by UNICEF and the Campbell collaboration, where we discuss child well-being interventions for low and medium income countries. So, what is there in our field in terms of this knowledge, accumulation and transfer efforts. You all know the One Health Knowledge Nexus, where we have communities of practice. I'm sure most of you have interacted. And know also the challenges here, that we have various levels of interaction. It's not easy to navigate because this is a huge field, and yet we cannot identify the primary data in it on its own. We have the One Health platform, originated from Germany. Our German colleagues, I'm sure, are quite familiar with that. This creates a communication space, not only across data, but across different needs. So we have they have a dedicated position for young scientists and also information for research funding. In Surveillance Codex document. Sorry, I thought somebody took the floor. Where we have a knowledge integration platform. Again, we have a lot of documents there, not only publications, but guidelines, policy briefs, etc. And another notion coming from across the pond, from the US, we have the OH Smart System, the One Health Systems Mapping and Analysis Resource Toolkit, which have been also used by other countries, where we have the possibility to identify challenges and try to tackle these through a very structured workflow through the toolkit. So this is also an important aspect. What I would like to pinpoint, and ending with a CABI initiative about One Health Knowledge Bag, which is the closer to knowledge transfer, is that all these different efforts and initiatives respond to the needs they were made for. So it is important to understand that we have no pure evidence map on One Health. It is challenging, it is very challenging to update, but of course it is important to understand how and when this tool will have its own thing. One of the projects that we are working on an evidence map is the EU WISH project. I know many of you are involved with that. So here we focus on wastewater surveillance. And here we are now working on an evidence map that accumulates all the studies that had some quantitative evidence of efficacy or effectiveness of wastewater surveillance. And this will be out as a product of EU WISH soon. So I'm following up and closing with the research roadmap considerations, which is the next step after a roadmap. What do we want? How can an evidence map be incorporated into this idea? The idea is that we have to organize then the output of the evidence map into communicating to the community and giving the scientists and all related stakeholders a tool to change, adjust, or reset their priorities. And this is a research roadmap. There's a UN specific methodology of how out of an evidence map you go and proceed into a research roadmap. It is not an easy feat. I'm just giving you the numbers to understand how many people, because this, as you can understand, requires wide consensus. And what I would like to focus on is the output, which is really fascinating. Those of you who have gone through UN commissioned roadmaps, you will see that they have three types of actions. It's a quick win, things that can happen quickly and make a difference. A best buy, where we have the middle term impact of an intervention or an action, and the game changers, as their name says, where we have actions that can, in the long run, change a field. And my final few slides are about the science strategies, which is important also in One Health. So we have different platforms here, different, so to say, attributes. We have the different things that are related to how we can approach this and how we can endorse that. The science of science, it's last, but for most of us in One Health, it's also very important. The knowledge mobilizations, where we have to use the research evidence in a meaningful way. The implementation, science, and the rapid learning systems are also important attributes of these efforts. And of course, I will end with the data infrastructure, which, as I discussed before, is quite important in understanding the whole notion of the effort. Handbooks and toolkits, we don't have to invent the wheel. We have to also understand that different disciplines have gone out into being endorsing a global approach. Migration is one of the scientific fields where people are very knowledgeable, very close to what we discuss in World Health. I have the privilege of collaborating again on an evidence map with the Columbia University on forced migration. And indeed, the similarities with the field of One Health are striking. And of course, migration is a component of One Health. And without, for the economy of time, I will stop here. And I would like to receive questions, if any.
Thank you very much. Are there any questions? I don't see. No? From the online? Okay, we don't have any questions. I have one.
Wonderful.
Okay, I'm not doctor, I'm a mathematician. I'm from a different field. So the question is, how can we collaborate better the different experts, the different domain experts in One Health?
This needs a structural approach. Thank you very much. This is a very important question. So we cannot do it by using the usual art of academic training, based on our intuition. There are structured efforts. The WHO has already put in place framework training, and I think there is already one collaborating center on One Health education. Where we have to train the trainer, so to say. We have to create that cross-disciplinarity mentality, which will help us understand how different aspects. So the good thing is that One Health is closely related to public health. And within public health, as you very well know, we are very diverse. We don't only have doctors, nurses, psychologists, mathematicians, architects, lawyers, whatever. So the field is most closely related to One Health has already accumulated experience on this interdisciplinarity so as to be able to go to transdisciplinarity. And I think that your project is an ideal platform to incorporate and implement initiatives such as this, where you cultivate a common mentality.
Thank you very much. So, thank you again. I would like to thank all the speakers for their excellent contribution and the audience for the active participation and questions. Thank you very much.
Thank you.
Thank you. Many thanks. Now, we will have to make the coffee break, if that's okay, slightly shorter, just to be on time and not create delays afterwards. So let's try to be back 11:30 or 11:35 at the latest, if possible. Thank you very much. with Professor Sotiris Nikolacheas that is chairing this session, and the floor is yours. Thank you.
Thank you. So, hello again. Welcome to this very interesting session. The first keynote about integrating spatial geometry and control charts for public health monitoring will be given by Professor Polychronis Economou from the Civil Engineering Department of University of Patras. So
Thank you. Thank you very much.
The floor is yours.
Thank you. Okay. Thank you for the introduction to the invitation. This is a joint work with Professor Thanassios Achlas from the University of Thessaly and Sotiris Bersimis of the University of Piraeus. It's on a published work on public health monitoring using control charts based on convex hull. The outline of my talk will be, I will give you a short introduction, a motivation, and then I'm going to focus on the monitoring method that we proposed. Finally, I'm going to conclude with an application with real data about health monitoring. Modern public health systems rely heavily on timely detection of any outbreak, emergency response systems, any abnormal patterns. And the problem is that nowadays we have to rely, it's not a problem, it's an opportunity actually, to rely on complex high volume data. We have data from sensors for emergency response system, digital platforms that feed us with an enormous amount of information and give us the opportunity to identify early warning signals in order to be proactive and make adjustments in the system. So this becomes more and more important and more challenging as more data are available. Traditional monitoring systems that comes back to 30 or 40 years ago typically examine indicators one at a time. We monitor the events of a disease. We monitor the incidents on the road, for example. We monitor simply single indicators. However, in real-world data, in real-world systems, abnormal patterns and extreme rare or public health phenomena almost never manifest themselves to a single variable. We have multiple factors that combine together, highlight an area at time as an outlier. So, what we need to do is to combine these signal changes in counts, in geometric distribution, in a user cluster in order to identify as early as possible any public health threat. And this also to support also timely decision-making and effective response. A tool to achieve this is the multivariate statistical process monitoring, a well-known statistical approach that enables early detection and, more importantly, also the reliable understanding of the key factors that drive this outbreak, this phenomena to behave abnormally. So, in this study, we introduced a novel monitoring procedure by integrated convex hull, a geometric feature that comes from mathematics, pure mathematics, and the chi-square-like statistics that from control charting techniques. And we are going to do that by monitoring simultaneously the number and the spatial distribution of events on a plane. Under assumption of an homogeneous Poisson process. I'm going to give you some definition also on these concepts as well. What is a convex hull? I don't know why this is not a very good slide, but okay. The convex hull is the minimum area that covers the convex hull is the minimum convex area like have a set of points. So at your left, you can see a convex area. At your right, it's not a convex. So we're going to use this concept, this geometric concept, in order to monitor the spatial distribution. Regarding the chi-square-like statistic, I assume that if not all of you, most of you are familiar with a chi-square test statistics, observed, mean expected, squared over the expected. Actually, this is an idea that you have an assumption, you expect under your assumption, you expect a certain number of events in this time frame or in this time framework. You observe something, your observation is very close to the expected number, everything is okay for your assumption. If not, this skyrockets and takes large values in order and give a signal that something is wrong. So based on this TIP concept, we are going to propose a monitoring system, algorithm, to monitor simultaneously the event, the counts of the events, and the spatial distribution of events. The motivation behind this can be found also in public health threats from infectious disease outbreaks, COVID-19, and other examples are very familiar, in which it's very important to have an early detection of outbreaks. And not only to detect when, but also where the outbreak was occurred. So it's not only necessary to monitor the time series of events, but also the spatial distribution. Another motivating example is emergency calls. This is actually our application. We have here the emergency calls 112 in Europe. You have an area that you receive calls of emergency, for example, health calls in an area. You expect to see more or less a uniform distribution of events, a constant rate of events, if every case goes under control. But if an event happens, you expect you are going to see probably a high concentration of the call at the peak, or end one of them. And the third motivating example that can be interesting to monitor the count of events and the spatial distribution is the environment monitoring, chemical and biological incidents. You need also to identify from where the pollution or the incident started initially. So you see there a higher concentration of events. So I can also give you more motivating example, but okay, let's move to the procedure of the algorithm. As I've already told you, we aim to monitor simultaneously the number and the spatial distribution of the event. And we are going to do that under the assumption that the event is consistent with a homogeneous Poisson process. This is not is a strict assumption, but it's not very unrealistic assumption. In order to give you what the formal definition, a semi-formal definition of homogeneous Poisson process is, I give you these slides. It's like you have a plane, you have an area. Under the assumption of a homogeneous Poisson process, the events occur in a time frame with intensity lambda events per day, per week, per year, whatever your time interval. Under these assumptions, the number of events in any sub-region of this area is proportional to its area. So if you are expecting the area of Patras, let's say 100 events per year, in the area of Frio, that's is the 10% of the area of Patras, you expect 10, more or less. Okay, this is assumption. Okay, and the other crucial assumption that given the events, given that you have a certain amount of events in this area, this actually, the distribution of this actual need, the condition of the number of events is a uniform distribution of the plane. So you have that all the sub-area more or less are proportional, the number of expected number of proportional to their area, and that condition to the number, you have a uniform distribution of the events on the plane. Okay, so As I already told you, we interested monitor two cases, the count and the spatial distribution. For the count of events, there's already a very powerful other control chart, the C chart, the count chart, the C stands for the count chart, that is actually built for a Poisson distribution. So we are going to use it as a benchmark for the monitoring the number of events in an area. The second proposed chart is a new one. Is our contribution, is a convex hull-based chart that monitors and detects any violation of the homogeneous opposed distribution assumption, meaning that aims to detect any abnormal distribution of the events in the plane. Let's start for the C chart. You have probably seen a C chart like that. You have the time point on the horizontal axis, the number of events in the vertical, and then every dot is the count of events at each day, second, or hour. You have an upper control limit. This is one side control limit because we are interested in a large number of events. If no disease is out there, you have a low number of events, everything is okay. So you have not a two classical two-side control chart, but one side. And this gray line, horizontal line, is the upper control limit, defined as the expected number plus 3 times the standard deviation of Poisson. So you have a phase one procedure that everything is under control. You have the overall mean of your past, and you use this control chart in order to monitor the future. You take from phase one the mean, the standard error, and you have the upper control limit. So you have here 1, 2, 3, 4 cases that the number of events exceed the upper control limit. This gives a signal alarm that maybe something larger than expected has occurred. This is a typical control chart that is very well known in the literature. The proposed monitoring control chart for the spatial distribution is rely on a chi-square like statistics. The idea, I'm going to give you here an animation, hopefully it works. Okay, probably it will. Let's give me a second to slow it down. I'm going to play this animation several times in order to focus. At your left, you have a normal, let's say, typical behavior of the distribution of the points. At the left, you have an area like that has a high concentration of the events. So you have two cases here. One is that you don't have everything is as expected. The other one is that you have an area here that has a larger number of events. Okay. The idea of the control chart is as follows. If we remove one by one the points, the furthest ones, the outer ones. Okay, let me pause here. Okay, you see that I have removed someone. I think it's 60 or 70 points here in both cases. So they have, we remove one by one from each cases. You see that, let's say you have an area of one, and we have removed 10 points from the other, so we have 90% of the original area. On the other hand, we have less, because there's a number of points concentrated. If you did this, etc., at the end, you are going to have a small area with a large number of points in there, and the change in the area on the right, sorry, it will be very small. So I'm going to return again back here. Actually, you are also identifying the area, the last point that remains is the area with the high concentrations. So let me go back. How? Μπορεί να μου βοηθήσετε. Πώς γυρνάω πίσω. Οκ, θα γυρίσω από εδώ.
I'm on the camera.
Το PDF.
Θέλει να ανοίξετε την
Όχι,
αναπαραγωγή.
ναι, όχι
Να φύγετε από την αναπαραγωγή.
Κλείσαμε.
Ναι.
Απλά δεν υπάρχει το PDF. Χάθηκε.
Όχι. Με το escape δεν γίνεται.
Από εδώ πέρα πάντως. Sorry.
Ναι.
Δεν φαίνεται το PDF. Okay. The idea here of this test statistics that you remove one by one. So you have here one. You have by removing one the area that is the more furthest. In this area you have one event. So this is the observed, and this is actually the expected area of this event. You have the original area and the difference of the area by removing one point. So you have 100, if you remove the L0 is what expected number of intensity in the area as a whole is 100, you remove one, this will be the 1% of the initial area, so this one will be 1 and this will be 0. So in your, I'm not going to give you again the animation, in your left point, this plot, this was more or less 0, these terms. On the other half, on the right, at the beginning, the expected number was larger, and the other at the end was smaller. So in any case, this we are going to, on the left, be very small, and on the right plot, we are going to have a very large. So actually, we can use this test statistics in order to identify problematic abnormal behavior. The problem here is that even if you have a chi-square-like test statistic, it's not a chi-square distributed. Why? Because they are not independent. So you have to simulate in order to identify the critical point. What is your upper control limit that will give you an alarm that something is not correct? So because we cannot find it theoretically, we have made some attempts, but it was very challenging. We went through simulation from different sample sizes. We've done different number of events. And we fit several different distributions for each cell. For each of these number of events in the plane, we have identified that for almost all the cases, the best distribution was the Box-Cox distribution. The Box-Cox distribution is given by this formula here. It has 4 parameters, mu, sigma, V, and tau, that controls median, scale, skewness, kurtosis distribution. So it's a very flexible distribution that can fit almost in every data set. So it's not very surprising that it was this highlighted. The surprising thing was that these parameters were fully identified more or less by NI. So you have the 4 parameters with respect to n, and we have used, I have not mentioned it, fractional polynomial procedure. It's a, let's say, unsupervised, more or less, in a modern term, a regression model to identify the optimal regression transformation of the covariance set. And the R square here is very extremely large, 99 plus, let's say. So Given this equation, because the problem is that we will not have exactly 25, 30, 35 events, we're going to have 32, 31. You can plug in the number of events that you observe in any given time, take this for the parameters, and identify this upper control limit. So the monitoring algorithm, in order to give it in steps, it is step one. At this time point, you have to record the number of events and the coordinates of the events, because you need also to monitor the spatial distribution. At the second step, you have to construct a C chart for monitoring the mean number of events. Given the number of events, observe the D value of the test statistic that we proposed earlier, and based on the number of events, calculate the upper control, the upper quantile that serves as upper control limit for the chart. Then, using both procedures, the C chart and the convex chart, determine the process under control. In order to clarify this procedure, I'm going to give you an application using real world data. This data, as I already told you, has to do with emergency medical calls to United States from a period of more than 5-6 years. In this small town here, in Montgomery, Pennsylvania, United States, and that they have a record. We have the timestamp, and because it's an emergency call, also the point, the GPS point of the emergency call. The problem here is that we had to first define the time interval, the time point, because it is an illustration application real data example. We have a time interval of 3 days as a sampling unit, so you have the calls aggregate as a time interval of 3 days, resulting to a 533 total 3 days time frames, approximately with 10 emergency medical tests per day. So you have 30 points there. At the left, you can see the C chart, and at the right, you can see the convex chart. You can see that a main difference between them is that the upper control limit on the C chart is a straight line. The upper control limit on the convex chart is not a straight line because it depends on the number of events. So you have not a straight line, but you have different control limits based on the number of events. Clearly, you have 1, 2, 3 events timed 3 time days that we have a large number of events, and we have several 1, 2, 3, 4, 5, if I count correctly, points that the distribution is not uniform as expected. In order to identify this, to visualize this better, we have extract 4 date intervals. The upper left map is actually the points from where the calls were made. We had not violation there, is the 3rd interval, so it's down here, let's say, and here, so everything is clear. We have an expected number, a standard expected number of events, more or less distributed in the whole area. The upper right, we have a large number of calls. So it's actually, if I recall correctly, this one here. So it's not very clear by eye, let's say, but actually there's a larger number of events there. The more interesting maps are the bottom 2, that are the 135 and 244. This is this one and this one here, where we have a clear abnormal behavior. And you can see here that there is a missing part here of calls. and high concentrated relative to the other areas here. And on the other area, you can see that here is a missing part, and probably here there's a more concentrated than expected concentration of coals. So, without plotting these events, we can actually identify that something is wrong, and then you can go back or at exactly that moment see what and where it happened. Of course, in a real case application, this will be more interesting to have a high frequency data, an hour, or in order to have no 3-day response. But if this case was actually an outbreak for disease, actually maybe serve also as a 3-day intervals time framework. More or less, thank you. That was all. Thank you for your attention. I'm open to questions.
Thank you. Any questions or remarks from the audience, please? Yes.
Hey, I enjoyed that quite a lot also because I could follow all the statistics, which I sometimes struggle with. I'm wondering regarding the application. You used like absolute 3-day windows that were following up on each other.
Yes.
Wouldn't it be far more accurate to get like temporal distributions to use a sliding window than an absolute distribution approach?
So you mean about using something like a rolling window approach or something like that?
Yeah.
Yes, that could be also a solution. But remember that we had already a problem here regarding the distribution of these statistics. So sharing also events in a rolling window, we are going to increase the distribution of this statistics even more complex. But yes, it can be done, but it has some more effort to do it.
Okay.
Thanks. But it's a possibility, yes.
Other questions or remarks? One by me. First of all, congratulations.
Thank you.
I appreciate a lot the rigorous approach you follow. Can you perhaps mention other potential applications of this work in the One Health domain?
Apart from public health outbreaks of diseases, etc., contamination of chemical and biological incidents. I have to think about that.
Yeah.
Probably there are many. Because,
but I believe it's quite broad. I mean, so...
Yes. Probably, I think that in animal control, I think that if you have events and spatial distribution,
Exactly. Yeah.
but there is actually all the cost of public health monitoring, you can do it.
Okay, thank you. Other questions or remarks? If not, thank you again.
Thank you.
And we continue with the second presentation. Another interesting theme by Mrs. Athanasia Paleologou, a PhD candidate here at the Department of Medicine, at the University of Patras. We will talk about AI and risk analysis in One Health.
Thank you all for being here today. As Professor Nicolas said, I'm Athanasia Paleologou. I'm a physicist, and today I'm going to discuss with you about the importance of risk assessment in public health, and I will try to approach as much as I can One Health. I'm going to show you two case studies. The first one, the most important, has to do with a case study regarding the quality of indoor air in a Greek hospital facility. And then I will move on with a second case study regarding risk assessment in swimming pools in Greece. I will move forward and very briefly with background regarding healthcare institutions and settings where it's very crucial to have a clearer image and assess indoor air quality, as we have to do with sensitive populations that are found there, doctors, patients, and general the public. In this case study, we're going to see how PMs, particulate matter in the rooms, in two rooms, virus and bacteria are linked. We're going actually to estimate probability functions, and we're going to see how all these pathogens are linked to chronic diseases, acute infections, and opportunistic bacterial infections by trying to quantify results. The basic objectives of this study are to assess carcinogenic and non-carcinogenic risks for all the pathogens that are mentioned above in two rooms, a doctor room in the hospital and an emergency room. We had actually an intervention in the room, so our probabilities have to do with pre and post intervention risk results after the intervention, which is a purifier, an air purifier, and then see how the influence of environmental factors such as temperature and relative humidity affect the parameters that are inputs in the model we're going to construct. The data that are gathered are particulate matter, specifically PM1, PM2.5, and PM10, that are gathered from continuous sensors within the room. Then the viruses that we assessed were SARS-CoV-2 and adenovirus from our laboratory, which are actually genome copies per liter. Then bacteria, where we don't have actually measurements, we have presence or absence. That was the only actual data that we had in our hands. And then we tried to link those parameters with environmental variables such as temperature and humidity. Regarding the methodology of carcinogenic risk assessment, we calculated the lifetime cancer risk by multiplying the equation of CDI, chronic daily intake, by considering the PM concentrations that we had from our sensors. We made some speculations regarding inhalation rate by literature. We assume the exposure time per day for people who are staying there, the exposure frequency, the exposure duration, and we assume a body weight of 70 kilograms for a typical adult. And then we multiplied this parameter, CDI, with the slope factor for each PM regarding the severity. For example, PM1 is more crucial to take into consideration because its diameter is a lot smaller, so it's very possible to for a person to inhale the matter, the particulate matter. Now, these are the slope factors that we assumed. The higher is for PM2.5, which is the most dangerous of all three. Also, we decided on a threshold regarding the PMs. I haven't mentioned it here, but it's one exposure to 1 million, which is actually a threshold that if it has surpassed, then we have to consider there is danger. And then regarding regarding the PMs that were modeled through logistic regression, we took those measurements and derived variables, constants actually, that had to do with the temperature and humidity in the rooms. They were measured with a different sensor, and we have our final measurements for each PM, which is actually the PM modeled through temperature distribution and humidity distribution. For non-carcinogenic risk assessment, which actually the virus and the bacteria, we had a viral exposure that we tried to estimate from SARS-CoV-2 adenovirus and from the bacterial colonies. The dose-response approach that was followed was something similar, like the PMs, but in this case we used a beta-Poisson function, where again we calculated the dose by assuming all the parameters that I assumed for the PMs, and then we also used a K, infection factor for each virus, for SARS-CoV-2 and adenovirus, to calculate the final risk of infection for each virus. Again, we try to consider K, the infection parameter, through temperature and humidity, as viruses are very affected by the environmental conditions within the room. And finally, for the bacteria, like I said, we didn't have measurements, so we had to make a qualitative risk analysis by using a score risk assessment, where we have, of course, presence and not absence. 0 is the absence and 1 is the presence, by assuming an LOD, limit of detection. It's actually 3 colonies that can be found in 100 mLs. This is the smallest colony of bacteria that we can assume that is dangerous for human health. And then we assume a risk weight and a sampling weight regarding whether a bacteria is a pathogen, an opportunistic pathogen, or a potentially pathogen. And of course, we had to assume a factor for the sampling weight, because it's different if we can derive bacteria from human skin, or from air filters, or from surfaces. This is a brief slide regarding the model assumptions. Like I said, all the environmental data were modeled in the model risk, the software that I use. It's an Excel-based, where I constructed the model, where I actually had temperature and relative humidity as log normal distributions within the model, so I can model the PMs and the viruses. Then I assumed all the exposure parameters that I used for calculating CDI, and all the rest are actually a summary of the previous slides. All inputs were incorporated through Monte Carlo simulations, 10,000 iterations, yes, in order to derive results, probabilities, and it was due to the huge number of iterations, we can easily capture uncertainty in our results. The outputs for each category are mean risks that we have within the program. For cancer risk and infection risk, where non-cancerogenic probability infections are estimated. And then we compared the pre-intervention and after-intervention results. In the first room, the doctor's room, there were 2 rooms, a doctor's room and an emergency room. In the doctor room, we can see that in the post phase, after the intervention, the air purifiers, the probability is a lot smaller. It's almost 15% decrease in the mean risk after intervention, which means that the model captures in an accurate way the decrease, and also the intervention works. That result was from PM1. Now for PM10, we can see an 8% decrease in mean risk cancer. All mean risk results are below the threshold we have assumed. Same for PM2.5 with a 9% decrease in mean risk cancer. Again, all our results are very low. A summary of this analysis, we have a decrease in our post-intervention results in the range of 9 to 15%. Regarding the emergency room, the decrease was 14%. Again, all probabilities are below, all risks are below the threshold from WHO. For PM10, a 7% decrease in the mean risk. Very low probabilities as well. For PM2.5, an 8% decrease in the mean risk. Again, all probabilities are, all mean risks are low. And again, there's a summary of the results regarding carcinogenic analysis. PM2.5 had an 8% decrease, and up to 14% decrease, with all probabilities being low. Regarding the non-carcinogenic analysis and the viruses, the emergency room, yes. The emergency room for adenovirus, the first day, the samplings took place for 2 days in the row for reasons of, I'm not sure about the word. Anyway, the sample had to taken place for 2 days in a row. So adenovirus and SARS-CoV-2 had very low possibilities. We can see that in the pre-intervention phase and the post-intervention phase, they're almost zero. Same for adenovirus and COVID-19. There is a very small decrease in COVID-19 in the post phase, but it could mean that someone was infected in the room and could transmit the virus. For the second day, again, all probabilities are very low, very close to zero. We have an increase in the post-intervention day on COVID-19 for the second day, probably for the same reason. And a summary of the results for day one, for adenovirus, we even detect anything in the room, all probabilities are zero. And for COVID-19, for both days, there was a slight higher risk in our probabilities. Regarding the bacteria, where we need to follow a quantitative risk analysis due to lack of results, for both rooms, doctor's room, actually this is the doctor's room, yeah, only the doctor's room, because we didn't have data from the emergency room. In both cases, pre and post period, the scores that the model gives to us are very small. For pathogens, of bacteria that are pathogens, it's only a 0.25 in both cases from human swamps. Which is, of course, very low. And for opportunistic pathogens and potentially pathogens, bacteria, all scores are very close to zero. And in both cases, we can see a decrease in the results. Also, there are some correlations from the model. Those correlation matrices are already part of the software that we used. It's interesting to see here, it's a bit chaotic, but I will try to highlight the main parts. There are very high correlations regarding PMs, PM10 and PM2.5, and also there were correlations regarding also high, almost 80%, 0.8, which is actually a high number, between viruses, specifically COVID and PMs, PM2.5 specifically. The results are here as well for the emergency room. Also, humidity and temperature played an important role in the risk estimations. We can see that humidity, for instance, has a negative effect, which means that if having increasing the humidity, the particles of the virus are becoming less. And it's not the same, of course, from temperature. We can see it possibly on the on the number 45, third line. It's again negatively, but with a lower, smaller correlation. Same for the emergency room. So that was a case study for the hospital. Now moving forward to a different case study regarding swimming pools and bacteria pathogens, where I used an algorithm using machine learning. The aim of study of this case study actually is how we can estimate the infection risk from Pseudomonas. Pseudomonas is a pathogen bacteria. And comparing the previous method, which is a Monte Carlo method with Monte Carlo simulations, with a Python-based algorithm. Then also we had the results regarding the sensitivity analysis, which parameters had the most important impact on the results, and then we will demonstrate how Python can reproduce those results. The data used there, actually the pathogen we want to examine is the Pseudomonas. It's a pathogen that is found in the water. We had also other measurements from E. coli, total coliforms, aerobic bacteria, and Staphylococcus. We had measurements from all swimming pools, and we used those bacteria as an index factor for the quality of the water of the swimming pools. We also used water quality parameters such as the chlorine, the pH, alkalinity, and temperature. We had also measurements of these parameters, and we assumed several exposure factors such as the water contact, the volume of the water that someone has on their body, someone that can ingest this specific volume, the time in the water, and we also assumed the buffers per hour. And again, when we used the model risk, 10,000 iterations. This slide where I say why we use Python, because in Python, in models, we can actually insert very easily the data, so the model can easily read and have results a lot easier and in less time than the Monte Carlo simulation. You can also compute risk statistics, and in this analysis, I try to estimate exceedance probability, also do the sensitivity analysis and see which parameters were the most important, something like a sensitivity analysis, for the results. This is how the model, actually, this is the Python code. We are not yet in the machine learning. The Python interface, this is how we insert through an Excel file our data. We have also assumed all the inputs that I mentioned in the previous slides regarding the risk analysis, which are actually inputs in our model. And after the model is run, we can see the correlation. It's not the correlation, it's the sensitivity analysis, of how all these inputs can have an impact in the probability of infection regarding the bacteria. We can tell that the chlorium is the most important one. It's on the right on our chart, which means that works as a protective parameter regarding the probability. We can tell the same about the Pseudomonas dose and aerobic dose, which actually increase the risk by remaining on the right part of the chart. This is how it is visualized, and then the correlations of all these parameters regarding the probability of infection from Pseudomonas. Chlorium is the most important one, the most possibly severe one, by contributing by 0.73 protectively again for the probability of infection. And then we used the machine learning algorithm. In this case, we used the random forest one, which actually a model where it uses several decision trees, I can tell, where all the models are gathered, and the most, let's say, accurate one, the most important one, is the final one where we can have our results. The model was trained and also evaluated. We can see the classification report here with using specific metrics. I have used a test size of 30%, which means that the remaining 70% is used to train the model. The random forest also produces sensitivity analysis regarding the importance of predictors, and one can tell that it's in accordance with the results that we took from Python. Chlorium, again, is the main factor that affects the result. Pseudomonas dose and aerobic dose the same as before. But also here we have the filtration rate and consumption of water, something we didn't see in the previous analysis, which are also playing an important role. And regarding the classification report, the metrics that have been used are the precision of the model, the F1 score, the recall. They're very high. The precision is almost 97%, the recall 99%, which means that the model gives accurate results and there's stability in them. A brief conclusion of this analysis. We saw how we can integrate model risk regarding risk analysis in frameworks such as risk analysis. It worked best for pre and post intervention, and we saw effectiveness and also accurate results regarding the probabilities. All PMs were below the regulatory thresholds from WHO. And also we saw that environmental factors played a major role in the results, but also in the analysis regarding PMs and viruses. And regarding the comparison of the methods, Monte Carlo, we can tell that provides a mechanistic, probabilistic estimation methodology, while random forest learned from patterns directly from the data. Of course, the result must be known. That's why it's a machine learning, a supervised machine learning method. But if the model can be trained, then it can predict other probabilities regarding the other case studies. Despite these different approaches, both methods agree on dominant risk drivers. This is, of course, it mentions the swimming pool risk analysis, because this is where I compared the two methods. That chlorine, Pseudomonas, and general microbial load are the key drivers. And also using both techniques, model risk and machine learning together, improves confidence in the findings, and also it can contribute in One Health. I know this is something that is focused on human, but it can also be used and utilized in several aspects of One Health approach. This is a brief discussion. It's not something that I didn't mention in the previous slides, so we'll pass it. And for future work scenarios, it would be best to have larger data sets. We can also use seasonality regarding the virus in such cases, or other data sets as well, different scenarios. it would be best if we do a quantitative risk analysis in bacteria, because we didn't have this data, we only had presence and absence. We could have information about the LOD and the weights, because all those were assumed, and we can also extend this analysis in other, not only healthcare scenarios, but in other settings, and One Health settings, of course. And that was it. That's some references, and thank you.
Thank you. Thank you for a very interesting and clear presentation. Any comments or questions? Please, go ahead.
Hi, I'm Magali from TU Dresden. Thank you for your nice talk. I have a question related to the first part. When you include the LOD for, you said it was around 3 CFUs, but I wonder if it's, or it make more sense to include the bacteria mechanisms of infection, because it's probably, depending on this, is gonna change if it's important that you have a very low bacterial at the beginning, if it's able to colonize or not. So maybe if it's able to reproduce it faster, so it doesn't matter if it's like few or more. So I think it's, I don't know if it makes sense to include the mechanisms of infection.
I try to input, to include actually this aspect through the sampling methods. We didn't have this data. We only had absence and presence of bacteria. I'm not sure if I can answer properly in your question. I'm not sure. It could make sense, but it's something that it's under construction, let's say. It means that we must be actually familiar with all the bacterial mechanisms. It could be done, but I can't answer properly because it's something that I haven't thought or considered yet, maybe due to the lack of the data that we had for this analysis. But yes, it could be considered. Why not?
Thanks for your presentation. You talked about in your future work about larger data sets.
Yes.
So for example, for the viruses, you saw this weird trend of like potential COVID infection of a certain person. So how large do you think a data set needs to be with regards to viral quantification to actually give you reliable results? How many days do you need of measurements?
I'm not an expert to answer this, but I can tell we could have, in my opinion, for a month maybe, to to make samplings actually in the specific room that we want to examine, and see how it goes. But I'm not an expert for this. I can't answer. It's only according to my speculation and my experience in the subject. Yeah. And in seasons, for instance, this sampling took place during winter. It should be done during the winter. Yeah. So we'll have to keep the seasonality aspect.
Thank you.
Just one. Uli, before we start doing that, we have seen the epidemiological trend in Europe, around Europe. What is the trend in different countries for the virus from the cases in previous years? And then we tried to have sampling in this very hot period of that, and usually we're doing 3 samplings, right? So to see more or less the average of viruses in every of these places. Of course, it tends to be very diverse because this virus is not very stable in all the cases. It has to do with the environmental conditions as well. For example, if you have humidity or everything. So I believe that in order to be sure to something like that, you need very big data in general to have it for this specific virus, not in general, because respiratory viruses are quite variant between them. So we are at the beginning of using this modeling on that.
Any other comment or question? If not, let us thank the speaker again. Now, last but obviously not least, we have Professor Dimitris Paraskevich from the medical department of University of Athens, a very well-known expert in Greece. I don't know, has the professor already connected? Okay. Hello there, please.
Good morning.
The floor is yours.
Me?
Yeah. Yes,
Okay.
we can hear you, we can see you. Please go ahead.
Okay. First of all, I would like to thank the organizing committee and especially professors from Patra, Professor Vandarakis and Dr. Anastasopoulou for kindly inviting me to participate to this conference. I apologize for not being able to be there in person, but since we had visitors from the ECDC and the National Public Health Organization, today it was not possible for me to be there in person. So I will share my presentation right now. Okay. So I hope now you can see my presentation. And the topic of our discussion will be about the relationship between wastewater viral load and clinical markers during a long period of Omicron subvariants in Greece. We are familiar with the situation of the COVID-19 and we all experience the social, the personal, the medical burden of the pandemic. And although the pandemic is not at the acute phase, the COVID-19, the SARS-CoV-2 virus continues to circulate, and therefore we need a reliable tool for surveillance and to quantify the viral load and the dispersal of the virus in the community. For this purpose, the wastewater-based epidemiology has been shown to be a very effective and a very powerful and cost-effective approach to communicate the community viral load. And the last benefit of the waste-based epidemiology is that the estimations do not depend on testing coverage. That is very low in our case, in our days, and also it's independent of symptoms. In our case, in Greece, the National Public Health Organization has established and still coordinates a national wastewater network for SARS-CoV-2 viral quantification that serves as a strategic component of Greece's public health surveillance. So this network is not only a network for research purposes, but it has been established and continues to act as the network of laboratories that provide data for the surveillance. This is the map of Greece and the location of the different laboratories and the sampling sites that contribute data and measurement to the national network of wastewater-based surveillance, and they are located across the countries in Thessaloniki, in Athens, in Patra, in Crete, in Ioannina, at several points in Greece. Okay, what was the rationale of our study? The rationale was related to the fact that it has been shown that the viral load correlates with reported cases with hospitalizations and deaths. And it has also been found previously that there is a lag time of approximately 1 or 2 weeks regarding the association of the viral load and intubations and deaths. So, but due to the fact that there were no studies about the relationship of the viral load and the disease severity or the disease burden, especially during the Omicron subvariants, we aimed to answer this question, and particularly we aimed to determine the correlation between the viral load and the clinical measures, including hospitalizations, the number of intubations, and the number of COVID-19 related deaths during a large period of the circulation of the different Omicron subvariants. We also aim to assess the differences in the ratio between the wastewater viral load and the clinical markers during a long time period, during approximately 3 years of the time of the circulation of the Omicron subvariants. Actually, we decided to use the ratio of the wastewater viral load to the clinical markers, since we were interested in the time periods when the burden of the disease, or the number of the clinical markers is increasing relative to the wastewater viral load. In our study methodology, we mentioned previously that we used measurements which were available from different cities, from different labs that cover the 70% of the national population in Greece. So we had an extensive and large coverage about the population of Greece, and the quantification of the viral load was performed at laboratories which were accredited, and they used a unified, harmonized protocol for the quantification of the SARS-CoV viral load, and that was very important in order to ensure methodological consistency. We also estimated a national measure, a national indicator that was weighted for the population from each city. And this makes, so we used a single national measure that was based on the measures from different cities weighted from the corresponding population size. For the purpose of our analysis, we used, I'm not planning to go very much into detail, but just to have an idea that we used distributed lag models to quantify the association, the relationship between the viral load and the different clinical markers, and also to estimate the time lag of these associations. And we performed also segmentation analysis to identify potential segments, potential time periods where the ratio of the viral load versus actually the ratio of the clinical markers and the viral load was different across the time period of our study. And particularly, we estimated three different ratios: admissions versus wastewater viral load, similarly for the intubations and the number of deaths. These were the three different ratios. And then we applied a segmentation, a tool for segmentation analysis that was based on the estimation of residuals of the square differences between the measurements and the mean value, and a method that applies least square to optimize the duration of its, or the length of its period for an estimation of an optimized, using an optimized criterion, the least square criterion. Actually, there are some more details about this method. I think I should mention that we choose a minimum length of its period to be of 6 weeks, since we thought that we decided that this is a reasonable period from a epidemiological perspective. We wouldn't be able to identify shorter period in duration, since we thought that it doesn't make sense, not reasonable to have very short periods. And we applied Bayesian information criterion for the optimization of the method to find which is the optimum combination of number of segments and the length of the segments. I can mention that in the Bayesian information criterion, there are two terms: a maximum likelihood term that finds which combination of segments fits better to the data, and also a penalization term that has to do with the number of the different segments. But I'm not planning to go very much into detail into the method. So again, I will skip the slide about the methodology. I will carry on with the results. So very briefly, we found that correlation between the wastewater viral load and the different clinical measures is very high for all of them. The coefficient of determination was higher than 0.75. For the new death was a little bit lower, but still very high. The optimal time lag, was 0 weeks for the hospitalizations, but it was 2 weeks for intubations and deaths. So as we can imagine, the intubations and deaths follow, and the optimum time lag is approximately 2 weeks regarding the measurements in the wastewater. These are some figures that show the correlation of the wastewater with the different 3 clinical markers. In the upper part of the figure, it's the correlation between the wastewater viral load and the number of hospitalizations without time lag. There is no need to have time lag in this correlation. The correlation, as you can see, is very high, and this is the case for all the 3 clinical markers. But the two figures at the bottom for the intubations and the number of deaths, they include a time lag of 2 weeks. So for all cases and for all the 3 clinical markers, as we can see, the correlation is very high. Now I move to the segmentation analysis and to the different time periods to the identification of the time periods with differences in the ratio, first between the number of admissions, the hospitalization, and the viral load. And this is the line, this is the results of this analysis that shows us the time trend of this ratio with the different segments as shown with the dashed vertical lines, and the horizontal black line corresponds to the mean value of the ratio within each period. As you can see, there are 8 different time periods with differences in the mean values of this ratio. The beginning, it's approximately moderate, and then as we move, we have a short period with a very high mean value, suggesting that the corresponding number of hospitalizations are very high regarding the viral load, or it is a period of a high hospitalization burden. We can interpret it in this way. And then we have a third period where the mean value is getting lower, or the corresponding number of hospitalizations are much lower regarding the viral load, the wastewater viral load, and so on. At the top of the figure, we show the different periods of the dominance of different subvariants. And if we look at this slide, it's important to note that, first of all, that we have periods with high ratios, low ratios, and moderate ratios. In general, the periods of the low ratios follow the periods of the high ratios, and also the different periods do not completely align with the periods of the dominance for different subvariants. This is, again, for the similar to the previous, but it shows the ratio and the corresponding wastewater viral load. We show in blue the ratio, between the hospitalizations and the wastewater viral load and the wastewater viral load alone. The periods of high ratios, more or less, at least after the middle point of the study period, correspond to periods of very low viral load circulation in the community. I continue, I will skip this one. I will continue with the ratio for between the intubations and the wastewater viral load. In this case, again, we identified 8 different segments. Again, we had high periods and low periods, which follow each other. And again, there is no perfect alignment. There's not very good alignment with the time period of dominance of the different subvariants. But this is one of the ratios for which we can observe some alignment, especially after the midpoint of our observations, especially after the midpoint, the middle of 2023. It seems that there is some alignment, there is some correlation of the different periods, of the periods with the differences in the mean values of the ratio and the time periods of the circulation of the different subvariants. It's the only measure, it's the only ratio that shows some correlation with the different periods of the subvariants. Again, the same figure that shows both of the measures, the ratio and the wastewater viral load. And I'll skip the slide. I will show the ratio of deaths versus the wastewater viral load. Also here we have 9 time periods, a similar number of periods, of segments with differences in the mean ratio. But the interesting and the important thing to consider here is that the mean value is declining over time. And if we look at the periods with the low ratios, for these periods, the mean value is also declining. The last one is lower compared to the third one from the last, or the 5th from the last, and so on. So if we look at the different segments with the high mean values, the ratio is declining. And this is also the case for the different periods, for different segments with low mean values of this ratio. So the ratio of the deaths versus the wastewater is steadily declining over time. And this is something important to keep in mind. I will skip the slide and I'll show the last one from the results. These are the results of the combined analysis of the 2 different ratios for the hospitalizations and deaths. For the hospitalizations shown in blue and the deaths is shown in red, and we show only the mean values for the different segments. So, and it's interesting to mention that the different segments for the lows and the high ratios, they align, they correlate very well. And these are shown in, they are highlighted in blue and orange, except the last time, the last 6 months, where the mean ratio of the hospitalizations was increased, and that was not the case for the ratio for the deaths. So if we combine all the ratios, the better alignment, better correlation was observed for the mean ratio of the hospitalizations and deaths. And importantly, the periods of the low mean values show very good alignment, except the beginning of the study period and the last 6 months of our study period. So in conclusion, the wastewater viral load continues to be a reliable measure about the the levels of the circulation of the virus, but it's also can be used as a predictive tool, especially for the severe disease and also death. We identified by segmentation analysis time periods where the mean value for the different clinical measures shows differences in the ratios. The segmentation patterns did not coincide in general with the time period of the dominance of the different subvariants. And the continuous patterns of the pattern of the continuous ups and downs indicate there are periods of increased severity, or the periods of increased severity follow the periods of lower severity, probably reflecting fluctuations in population level immunity. One potential explanation for this observation could be the differences in vaccination coverage, but as the vaccination coverage is very low after 2021, we think that this pattern probably is due to the differences in the population level immunity. Importantly, an analysis mentioned before, the ratio for the COVID-19 deaths versus the viral load shows a steady decline over time. And this seems to be a potential reason for that is the better management, the availability of antiviral treatments, also the higher levels of immunity in the population and the continuous exposure to the different subvariants. And final conclusion is that, this has been discussed. Final conclusion is that we show clear evidence that even in the case of the circulation of Omicron that has different clinical and virological characteristics, and in the period of long circulation of different subvariants, the burden continues to be significant. The number of hospitalizations, intubations, and death continues to be important for public health reasons, although the ratio of deaths is decreasing for the reasons that we discussed. And also the association between segmentation trends and the dominance period seems not to be significant over time. So thank you very much for your attention. That was a brief overview of the study that we performed in collaboration with all the centers that contribute to the national coalition about the wastewater-based epidemiology for SARS-CoV-2 in Greece.
Thank you. Thank you, Professor Paraskevis, for a very insightful presentation. So any comments or questions from the audience? Maybe a question by myself. Do you think that the complementary use of machine learning methods could provide some additional inference about your findings?
Yes, I fully agree, especially if you want to investigate what are the potential factors that correlate with our findings, especially if we want to provide evidence of what causes these ups and downs in these ratios. Okay. In these ratios, yes, I fully agree that machine learning can give us an insight, and it can be very useful in addition to what we have seen as to get an initial idea about the patterns during the time period of our study.
Right.
The answer is certainly yes.
Yeah. If I may, because in some cases the phenomenon is so complex that rigorous statistics cannot really capture the non-linearities and the multiple relations. So any other?
Yes, I fully agree.
Yeah, thank you. Any other question or remark? I guess everybody is hungry now, so I'm going to break for lunch. Thank you again, Professor Paraskevis.
Okay, thank you. Thank you very much about the invitation.
Okay, thank you. So time for lunch.
So thank you for being back here with us. We are ready to start session 3 of day 4. We will have a very interesting roundtable with a title Artificial General Intelligence for Strategic Planning and Decision Support. And since Professor Polychronis Economou will chair this session, I will immediately give the floor to him to continue. Thank you.
Okay, thank you for the introduction. Welcome all to this session that starts with a roundtable and has additional 2 keynote talks. The first part is the roundtable with 3 colleagues from the University of Thessaly on artificial general intelligence for strategic planning and decision support. The speakers were going to present a 10-15-minute talk each on methodology, application, and policy making and application, let's say. And then we are going to have an open discussion with questions addressing to all of them. So the start is the first talk going to begin by Professor Kouvrekis Ioannis. Kouvrekis, okay. So the floor is yours.
Thank you very much. Let me find the full screen.
Yeah, yes, but is it shared? No. Wait.
Using the share screen.
Yeah. Is it that one?
Yeah. Okay.
Something like that. Αυτό? Okay.
Okay. Generally, when we want to talk about what artificial intelligence is, we present several examples from different areas. Of course, there is a huge, how to say, we live in the AI era, okay, but when we talk about AI, we don't talk about AI. We talk about application of AI. So I will start with what artificial intelligence is not. Artificial intelligence is not a supernatural identity which can kill us, okay? It's not the Schwarzenegger, it's not a robot like that, because the majority of people believe that AI is something like that. But AI is just mathematics, a lot of mathematics. The first wave of AI consists of symbolic AI, reasoning, rules, which was a rule-based AI. The second wave, which is the machine learning wave, is another branch of AI, which try to solve 2 fundamental problems in supervision, in supervised machine learning era. We try to solve 2 different and similar problems, the classification and regression problems. Classification problems is when we try to find the label of some objects. And regression is a similar problem, but we want to find the value of these mathematical objects. In more abstract level, the machine learning, the supervised machine learning era consists of one fundamental problem: to find an estimator, a function which simulates an unknown function. We try to find a target function. The target function which assigns pictures to labels, features to values. We don't know this target value. That's why we try, using ML techniques, methods, to find another function, the predictor, the classifier, for example, which is another function which simulates the target. Okay. And how we solve these problems? We're using two steps. The first step is the model selection, and the second step is model assessment. This is one of a lot of different workflows in a machine learning problem. Okay, here's another workflow. In the model selection step, we have a lot of model assessments, sub-steps. And the problem is that in One Health, we have the problem of translation, which is the same problem, different teams will have, in the same problem, different teams will have different results. Why? First of all, we have a lot of common issues, like overfitting. Overfitting is when our model learns specific patterns from training data, but we have very low, poor performance, poor accuracy in unseen examples, in unseen data. We have the other fitting problem, which is when we cannot find an appropriate model in order to solve our solution. Okay. These two belongs to an umbrella term, the pool generalization. We can have a generalization in our problems. Also, the selection of the features is very important because with noisy and irrelevant features, we will have a very poor accuracy in our problem. And as I said before, for the same problem, different teams can have different solutions. Why? Because they don't know that different features for the same problem, imagine that I want to find a predictor for cancer or for find if this picture is a dog or a cat. Using different features, fundamentally, you have different problems to solve. And this is the problem of translation. Because in One Health, we have domain experts from medicine, environmental science, mathematics, and we must find a universal language in order to translate and transform the problems from one field to another. Also, the lack of translation yields to the lack of explainability. We want explainability. We want to know which features is very important to our results. Okay. So the solution is something that it happens into our department. Our department is Department of Public and One Health in Thessaly, University of Thessaly. And there we are at least 3, 4 scientists from different fields. And this team has tried, and I think that we have some very good results, in order to find solution in machine learning, using machine learning methods, in order to find solution in medicine, environmental problems, and what other? Yes. Now, Professor Angeliki Katsafadou, she will present you the solutions, the problems and the solutions. You will see from different areas a lot of problems. And in the workshop, in 2 hours, we will see how we can avoid all these technical issues in ML projects, problems. Okay? So.
Thank you. Let's move to the second speaker, Professor Katsafadou.
Good afternoon. Okay. Good afternoon, everyone. Thank you for the invitation. It is a pleasure to be here. My name is Angeliki Katsafadou, and I am an associate professor at the Faculty of Public and One Health at the University of Thessaly, and a Fulbright alumna from the Yale School of Public Health from USA. My presentation today, I will discuss how AI can help transform laboratory data into actionable predictive intelligence. As the previous speakers have discussed about so much, the One Health approach is a 21st century framework that recognizes the deep interdependence of human, animal, and environmental health. While the term itself is quite recent, the idea has long been recognized, and its importance was emphasized during COVID-19 pandemic. One Health aims to address complex global challenges like zoonotic diseases, antimicrobial resistance, climate change, and pollution through integrated collaborative solutions. It reminds us that protecting one area of health requires working across all 3 systems: people, animals, and ecosystems. As One Health challenges grow more complex, the need for digital innovation is clear. Pandemics, antimicrobial resistance, and climate-driven risks are evolving quickly and interacting across human, animal, and environmental systems, faster than the traditional surveillance can track. At the same time, laboratory data are expanding rapidly from microbiology and genomics to environmental measurements. This creates enormous opportunity, but also demands better tools to interpret high volume and high variety of data. A major barrier is data fragmentation. Human, veterinary, and environmental datasets often remain isolated, limiting our ability to see cross-sector patterns. This is where AI becomes transformative. By linking diverse data streams and detecting early signals, AI provides unified predictive insight that supports earlier detection, risk forecasting, and more coordinated One Health action. AI can support One Health in three main ways. First, it can accelerate pathogen and biomarker detection by rapidly analyzing lab results, genomic data, and sensor outputs, enabling earlier diagnosis and stronger outbreak preparedness. Second, it offers predictive analytics that forecast disease and environmental risks based on climate, animal movement, antimicrobial use, or ecosystem changes. This shifts us from reacting to crises to preventing them. Third, AI provides explainable models that support transparent, trusted decision across human, animal, and environmental health sectors. To turn, though, from laboratory data into predictive intelligence for One Health, we first need standardized laboratory and genomic workflows across human, animal, and environmental sectors. Shared metadata and quality controls ensure AI receives a clean and compatible inputs. Next, cross-sector data integration platforms must link laboratory, clinical, ecological, and antimicrobial use data through the integrated systems and shared ontologies. This connectivity enables true cross-domain prediction. Finally, AI has to be deployed in operational decision systems. When AI outputs fit directly into clinical, veterinary, and public health workflows with clear explainability and validation, they support timely and coordinated action. In order to pursue the strategic planning, however, integrated digital platforms built on shared standards need to exist, allowing human, animal, and environmental systems to exchange data seamlessly. Governance must develop in parallel, setting clear rules for data stewardship, ethical oversight, accountability, and equitable access. AI tools should operate within a coordinated One Digital Health ecosystem, integrated into validated workflows for surveillance, diagnostics, and decision support. Cross-sector pilots provide the final proving ground, allowing teams to test workflows, evaluate fairness and accuracy, and refine governance and demonstrate impact before scaling up. I will proceed with some examples. In zoonosis, AI supports the spillovers prediction, for example, by integrating ecological, climate, mobility, and livestock data to model conditions that elevate the zoonotic risk. Use of AI on genomic data that are abundant, for example, help identify mutations or host range shifts that suggest pathogens which may be adapting toward human infection. By combining the environmental and contact pattern data, AI also maps high-risk human-animal interfaces such as deforestation, fronts, or wildlife livestock zones. AI-driven deviation reveals unusual signals like unexpected wildlife morbidity or shifts in pathogens diversity, proving early warning alerts. Together, these capabilities build on a more integrated One Health intelligence system that link the ecosystem, animal and human data for earlier detection and response. AI can be used also to support pathogen identification. Here is an example for a project done in our faculty showing how AI can help the rapid bacterial identification and antimicrobial resistance control. We used publicly available Raman spectroscopy datasets from 30 important pathogens. The spectral data were cleaned and normalized in order to remove the noise and keep only the useful information. Then machine learning models were trained to classify the bacterial species based on these spectra. To understand how the models made their decisions, we applied explainable AI methods that highlight which parts of the spectrum contributed to its prediction. With this workflow, we achieved fast and accurate bacterial identification without the needs for previous cultures. This shows how AI combined with advanced lab tools that modernized traditional microbiology and speed up the diagnostics. Also, AI supports antimicrobial resistance control in several practical ways. First, deep learning models can detect AMR genes directly from wastewater environmental metagenomes, for example. giving us an early view of resistance circulating outside clinical settings. Second, AI helps identify the key drivers of AMR spread, such as plasmid mobility, gene transfer events, and antimicrobial use patterns, so that we can target interventions more effectively. Third, by combining genomic data with growth curve or other omics information, AI can speed up antimicrobial susceptibility prediction, reduce the time needed to determine which drugs are likely to be effective. And finally, AI-enabled stewardship tools can guide antibiotic choice and dosing at the point of care, supporting more precise and responsible antimicrobial use. Regarding milk quality and safety, we demonstrated in another study how explainable machine learning can support milk quality management in sheep and goat farms. We used routine bulk tank measurements like somatic cell counts, total bacterial counts, pH, and fat-to-protein ratios together with farm management and climate data to predict good versus poor milk quality in the bulk tank of farms. We trained multiple machine learning models and applied explainable AI tools like the SHAP values to identify which factors most strongly drive quality changes. The somatic cell count and the total bacterial count emerged as the strongest predictors, with climate-linked variables also influencing the outcomes. By making the model's reasoning transparent, this approach helps farms understand which risk factors and supports more informed decision-making for food safety and quality. Further, in a new work, in addition to bulk tank quality prediction, we also applied machine learning to support mastitis control in dairy sheep, because subclinical mastitis is a major hidden driver of reduced milk quality. Using data from 267 farms covering management practices, flow characteristics, and environmental variables, we trained models to predict subclinical mastitis prevalence. The analysis showed that hygiene conditions and milking practices were the strongest predictors, highlighting actionable areas for improving flock health and protecting the milk quality. Further, we conducted a complementary bibliometric analysis to examine how machine learning and AI are being used for mastitis control in general. We found only 122 studies that were retrieved from PubMed and Web of Science, and we found applications ranging from sensor-based early detection to image-based diagnosis and treatment decision support. However, the review also exposed some key gaps: limited use of explainable methods, reliance on small datasets, and very few studies on small ruminants. These gaps underscore the need for more transparent, robust, and One Health aligned AI tools in this area. Our teams also explored the use of explainable AI for detecting and characterizing microplastics in environmental samples. In this study, Raman spectroscopy was combined with explainable AI methods to rapidly and automatically identify different types of microplastics. The integration of explainable AI not only improved classification accuracy, but also made the decision process transparent, allowing us to understand which spectral features drive each prediction. Another environmental application of our group is assessing global exposure to gas flaring. In a recent study published last week, we produced the first worldwide estimate of how many people lived near upstream flaring sites from 2016 to 2023. By combining satellite observation with global population datasets, we showed that millions of people are exposed, with most of the impact concentrated in a small number of countries. In some areas, exposure actually increased, even though flaring volumes went down, simply because flaring activity moved closer to where people live. We also generate projections up to 2030 to highlight where population exposure is expected to rise in the coming years. Linking lab data with climate intelligence is not just about adding environmental conditions. It involves modeling how pathogens behave under specific climate variables, such as temperature-dependent growth rates, humidity-driven survival, or vector suitability shifts. We can then connect genomic or laboratory-confirmed pathogen detections with concrete climate exposures, such as heat waves, rainfall anomalies, land use change patterns, or seasonal climate cycles. This creates a data-driven understanding on when and where environmental conditions amplify risk. Finally, climate pathogen models allow One Health teams to design climate-adaptive strategies, issuing early warnings for vector-borne spillover, mapping livestock disease risk under projected climate scenarios, or setting environmental hazard thresholds to trigger surveillance. This is how lab data and climate intelligence work together in a practical, operational One Health system. However, as we advance AI in One Health, we have several key challenges that need to be addressed. First, there is a need for harmonized data standards across human, animal, and environmental health systems. Without system integration, we risk fragmenting datasets that limit the accuracy and scalability of AI models. Second, explainability in AI decision-making remains essential. Public health and clinical users must understand why models generate particular predictions and to try to modify if they wish to do so. Finally, sustained cross-sector and transdisciplinary partnerships are crucial and frequently lacking. These partnerships are needed to ensure that AI tools are relevant, ethical, and aligned with the real-world needs. As we move forward from challenges to the future, or to tomorrow's capabilities, AI enables us to build a more proactive and predictive One Health system. First, AI allows us to transform raw laboratory and surveillance data into actionable intelligence. Instead of isolated data points, we gain meaningful insights that support faster and more informed decision-making. Second, AI enables the creation of predictive dashboards that integrate information from humans, animals, and the environment. These dashboards help us identify emerging risks earlier and understand how different domains influence each other. Third, this leads to a fundamental shift from reacting to crises to preventing them. Preventive models help us intervene earlier, reduce disease burden, and optimize the resource use. Next, the real-time monitoring platforms strengthen situational awareness. They allow us to track changes as they happen, whether in pathogen trends, environmental exposures, or antimicrobial resistant patterns. Finally, all these components contribute to building a truly real-time preventive and systemic One Health approach. This is the direction we need to move toward if we want to address complex threats effectively. And the center of all this is collaboration. None of these advances are possible without strong partnerships across sectors: public health, veterinary medicine, data science, environmental science, and policymakers. Collaboration is the key. Thank you very much for your attention.
Thank you, Professor Katsafadou.
Thank you so much. I think the question is
at the end.
after.
Okay, yes.
In this QR code is my profile on LinkedIn if you wish to connect. Thank you.
Okay, thank you. Let's move to the third speaker of the roundtable, Professor Diamandopoulos. Professor, the floor is yours.
Okay. Dear and honorable committee participants, now is my time. First of all, I warmly thank the committee and the organized committee, and especially Professor Apostolos Bantarakis, with whom at the past I have closely collaborated when I was General Director of Region of Peloponnese, and especially and the President Professor Economou for the honorary invitation. My special object is the policies of health, strategies, policies, and administration. And many times the policies is a little, they want to have conclusion in the One Health or the total health, but conclusion is confusion many times. So I try to think about Internet of Things and with AI and how it is possible, okay, to connect. Before 3 or 4 days, excuse me, yes, here we are. I saw a biography of Mary Shelley. And this is, I think, the modern dilemma of Prometheus. This is out of my presentation. And the modern dilemma of Prometheus is AI and the future of, or different, Frankenstein and the future of AI. And believe me, this is strange, but because a monster made by many. So this is opportunity for the human people and the human being anywhere to change this rule. This is about health. World Health Organizations, the absence of any disease of equality, the state of balance that person has created within themselves and between their social and natural environment. You see the rules, the doctors, the patients. Many times we have the difference, what is illness and what is disease, because the part of doctor and the part of illness, they are different, the patient vs doctor. So we see many different risk or suffers or things that we must have, because what is health? For me, health is this that we say in the public health, social health, mental health, and health. But in One Health, we say that we have 3 cycles: the cycle of environment, animals, and people, humans. These 6, different cycles, this that we call public health and One Health, is an opportunity. And I think this is the strategy for AI. Many times I talk a lot, and many times I talk very little. We'll see. And forgive me because I have a little tiredness because I was in another related conference. Now we see, because I don't want to have a lot of time, primary, secondary, and tertiary health. Different levels of health and health services, and with different challenges. I want primary, and primary improve access to healthcare systems, and a visit to the main therapist, doctors, nurses, and physiotherapists. The other levels, they are more difficult. It has to do with therapy and not with diagnosis models or with methods that we want diagnosis-related group in preventive, not in therapist. So we want to change, and to have power in the primary, and AI maybe help these models. This is the cardiovascular various forms of cancer, CA, COVID-19 pandemic. We see the main causes of death 2020 in the Middle East, the Europe score. Greece is not very good, but close, and has the different these different types, okay, categories. And now we have the Internet of Things because we must work in these models. An ecosystem consigning correlated parts of elements. This part of elements, you see, is a cycle. I like cycles. These part elements intersect with each other at local, national, or global level. And you see in the left the end user, the devices, the application environment, the relevant professionals, and the developer. They are applied in fields such as, you know, urban environment, resident environment, entertainment, public transportation industry. In the back, you see the industry and the cloud and the automized. But we want to say about MIoT, Medical Internet of Things. Very quickly, a network of devices such as sensors that interact with each other in a diverse network. This is the definition. The evaluation is by the end of 2025 is the estimate that 1 trillion such devices will be in use. We see then the data collection, the recommendation, end users, sensory devices, communication system, environment application, and of course the recommendation. Health professional, patients or participants, regulatory authority, device, etc. Internet of Things, possible solution for synchronous and asynchronous health monitoring, easier implementation and monitoring, reduced operating cost, but early stages of development, several challenges regarding their adoption and risk or unsuccessful implementations among the population. Okay, you see, and now this is good because the monitoring the health of the population we want for to protect, okay, and we have a governance privileges and need that arise. So the artificial intelligence goes to diagnosis and treatment, and at last health system, because we want smart living networks, high levels of patient care, and reduced cost and time per patient, because in the economics of health is very important. Challenges and opportunities, okay. Practical application. We want artificial intelligence, okay, machine learning, specialized knowledge of various systems, and techniques for reproducing and disseminating knowledge. And MIOTs, I have a lot of different pictures, and I want to be more focused. MIOTs are still in the development stage. In this context, challenges and difficulties arise in relation to the following we see here. Challenges. Restrained access of healthcare professional certifications, incomplete access to certificates and training seminars, and you see the conclusions and the cases. The limited cases and specialized of healthcare professional has a negative impact on the reality of SLN, and the lack of certification and the use of smart devices could negatively affect the implementations. Cases healthcare professionals need feedback with new information to the successful implementation. Here, you see the cases, smart devices manufacturers need to conduct comprehensive researches on managing challenges, because if you see at the left side, and especially the 5, the non-application of bioethical rules. And here, the 7, the inability to upgrade the software. And if we see the cases, smart devices manufacturers need to conduct comprehensive research on managing challenges. These challenges could negatively impact the SLRS. And here, the case is a huge volume of data, okay, and these challenges could have negative impacts on the entire ecosystems. I don't know my time. Okay, so Mr. President told me about this. Okay, because
Okay, a couple of minutes, 3 max.
okay, here challenges lack of trust, patients who do not adopt or patients who find learning difficult, and conclusions lack of confidence in smart devices may be negatively affect, and patients who do not use smart devices or effectively could negatively affect, and the cases is patients and the end users. And here is the same about the lack of security mechanism and for defective massive use to ITO devices. Many other challenges, security issues, incomplete processing of information, lack of reliable connectivity, and the cases is the nectar structures is the most important party of interest for the implementation of smart living networks. Conclusions from this population health and formulation of future health and prevention policies. There is, and there will be continue to be an inextricable link between the use of technology and assessment and provision of health for the entire population, regardless of social, economic, endogenous or environmental factors. Whatever definition of health is adopted, especially the third one, due to the enormous challenges facing societies, the application of technologies and smart living networks aims and will continue to aim at improving the health of the population. The design and as acceptables are of exceptional and indisputable importance. And the application of health is multi-layers because the complexity and types of pathological conditions treated by health professionals, special and complex and specialized for the production required and specialized equipment and space. Each level of healthcare provision based on the above and has its own unique or common characteristics. This has English and Greek together, but the application of health is multi-layer, while the good and the purpose are one. Okay, and these are conclusions about the cases and the challenges, but the three most common causes of death among the European population are related, okay, to cardiovascular disease, various forms of cancer, and the COVID-19 pandemic. I think that the CA maybe after 3, 4, 5 years is the first, in Europe. And smart living networks and IoT have similarities and differences, which are smart living networks are a specific type of IoT. Smart living networks aim to develop target, sophisticated and intelligent systems that can be applied in living environment, and Internet of Things refers to a broader network of devices and systems that are connected to each other. It's applied in the field of industry, transportation, and other sectors. An example, this is a program. I have only 4, I think, pictures. This is a program, a Smart Bridge, a Horizon program. I have the honor to be PL Sign, a public leader in this program in Greece. There are 6 or 7 different countries of Europe, and I was clinical director in the region of Peloponnese. With different challenges and promoting health and independent living of the elderly. You see different types, balance disorders, cardiovascular frailty. And this was a big innovative platform, evidence-based, with big data analysis, security and safety, active recommendations, integrated care, active monitoring, support at home. This has to do with technical objectives and clinical objectives. With a big data, and evidence-based personalized support. And this platform, continuous and objective monitoring, intervention for personalized medicine forwards, optimizing disease, measure improvements to the quality of life and elderly and their ability to live independently. This is a program in the region of Peloponnese for 5 years. And there are, I think, 5 or 6 different municipalities, the municipality of Paleofaliro in Athens and a municipality in Thessaly, Trikala, and 3 municipalities in Peloponnese, Tripolis, Sparti, and Kalamata. This is okay to promote health and independent living of the elderly, and we try to have results and big data. But when the programs arise or stop, unfortunately stop in the data and the big data, because all these systems want a future and money. So thank you a lot, okay, for time and attention.
Okay, let's thank all the 3 speakers for the introduction to what is and what is not the AI, their application, and the opportunities and challenges the health ecosystem. So we have 10 or more minutes for an open discussion, questions, and remarks and comments. So, yes.
Thanks for those insightful talks. So, for me, this entire aspect of explainable AI or explainable machine learning, not sure which of the 2 terms is the correct one, is quite good, because I think we as humans learn from mistakes, and in the non-explainable machine learning aspects, it was difficult to identify what are the mistakes that were made when you improved something. And so I think I've got 2 questions regarding that. The first one would be, how easy is it now if I realize I need to retrain the model? Do I use the old model and tell me what was the problem in the old one so that I can learn from it? Or is it just a complete new setup recalibration? And the second question would be, does integrating explainability come with trade-offs for accuracy compared to non-explainable machine learning aspects? And if that's the case, where do we draw the ethical line between I want to know what's going on and I want to see that it's as accurate as possible to help health? Because I feel like with health, it's always difficult to make these ethical trade-offs.
Okay, thank you.
Yes, it's a huge answer. Look, explainability is an umbrella term because artificial intelligence is not just a small field. It's a meta field. AI consists of mathematics, computer science, optimization problems, etc., etc., etc. The problem is that we must define what we mean explainability. Okay. It's not easy answer. The most common techniques now use not statistical methods, but game theory methods. And based on these methodologies, we try to see which features are important to the answer. And this is irrelevant to the ethical issues in ML, in machine learning problems. Now, for your first question about the retraining, etc., depends on the problem. I'm sorry, but I cannot give easy answer because there are not. Okay. It's very simple to define what supervised a classification problem is, but it's very difficult to give a sound and complete answer in a lot of real-world problems. I believe that we are in initial stage of explainability. I don't know if you're happy with my answer, but yes.
No, I think, at least regarding the ethics part, I think that explained it quite nicely.
Do you have your ChatGPT on your phone?
Probably, yeah.
Okay. Try to ask ChatGPT to draw an analog clock showing 6 o'clock. If you try now, probably, or a random time, okay, probably the output will be a specific hour. Try. Yes, it's 10 to 10. If you ask ChatGPT to draw an analog clock with a random hour time, probably the answer for all of the audience will be the same. A clock which shows 10 past 10, or 10 to 10. Why? Can you imagine why? Because the data from the internet, the majority of the images of clocks from the internet show this time. So the ethical issues in machine learning problems arise from the data, not from the algorithms. It's a huge misunderstanding. The algorithms is just mathematical procedures. The problem is the data, is the input data. If you feed your algorithm with, how to say, not appropriate data, you will have not appropriate answers.
And to add something to that, you have to be careful what questions or what problems, I think, what questions do you put to the model to answer? Because the model does not make assumptions, that makes only calculations. And calculation regarding the variables that you put inside the model, and with what's called the variety, the variety on variables.
The weight.
The weight of the variables, you set the weight of the variables by your biological knowledge. So, yeah, the explainability is starting from the question and from the biological question that you need to solve.
Right.
Because it just searches for images.
Because you said 6 a.m., I think, and I said like a random time, and it's both 10:10.
I know. This is a very nice, not bug. It's not a bug.
Okay, he got sick, but you pay for it.
It's an analog. Okay, but it's not a bug. It's not a bug. It's how machine learning algorithms work. Okay, it's not a paradox. It seems to us as a paradox, but it's not a paradox.
Okay, any other questions, remarks, comments? Okay, yes. Okay, just to elaborate more on this on explainable AI, I have a saying that mathematicians and engineers, because machine learning AI comes from engineering departments, have a lot of common, but some main differences. We, mathematicians, focus on theory, assumptions, strict questions. We invented the cycle. Engineers invented the wheel, and they realized that it can work even the flat. They can put it in the car and work and rotate even flat. So they make working, and the second difference is the branding. We have terrible names. They have artificial intelligence machine learning. Excellent names. So the road to explainable AI, it's one-way ticket. You can go back. We are going there because the mathematicians are on board on AI now. So we have the tendency to explain everything. So there's no way back. We're going to arrive there.
So are you a mathematician?
Yes.
Oh, we are majority here. Yes, mathematicians, we are terrible to branding. We cannot.
Yes.
I have a question related to, well, all these machine learning depends on data for training. Those data belongs to, related to the past, most of them. But in the context of a organism, bacteria, virus, evolve super fast. How accurate are still those models, or how do they deal with this rapid evolution, for example?
In mathematics, we don't have time. Yes, we don't have time. We don't try to forecast something. In ML projects, we try to estimate, not forecast. Forecasting is something different. We try to estimate. Now, you said that we have old version of data. In my presentation, I said that the same question, for example, I have a set of pictures, and I want to pictures of dog or cat, okay? And I want to find a model in order to predict if a picture is from a cat or from a dog, okay? If I change the number of the pixels, the question is the same. The problem is different. I have different number of pixels. It means that I have different number of features. That's why I said that it's very important to find a universal language between ML groups. So this problem that you refer is, how to say it in English, egenis, it's fundamental. It's in every problem. As I said, if you change the pictures, the pixels, you change the problem. Not the question, but the problem. I don't know if I answered to your question.
Let me add something. I will add with an example. In the projects that I previously presented, we had bulk tank milk samples which used for the machine learning analysis. But these were samples, let's say, from a 2 years project. In order afterwards to train the machine learning and check if the conclusion is correct, we went again to the field and gathered some new samples in order to check our results if they match with the current sample. So when we are taking the models and say, this is the best model, this works, and these are the outcomes, we can again go to the field or to the lab and take some new results, and then we do the analysis again with a smaller sample, with smaller data set, and we can do some overfitting if the results are quite changed. So we can predict, let's say, the biological evolution of this kind of organisms, and we can add them in order to have these different pictures that the mathematicians say, and do the fine-tuning in the models. We can do that.
We must understand that in ML, in machine learning, we try to estimate. We do estimations. So if the space of features is changing, it's no problem for us. Okay.
Okay. If there is no other question, we can proceed to the back again with the speakers. Thank you. And proceed to the first keynote speaker of this session, Professor Milan Stenlik. Milan, are you here? Are you online?
Hello, hi, Polychronid, how are you?
Hi, Milan, nice to meet you.
Am I audible?
Yes. We
Perfect.
can see your screen, we can hear you. You can just full screen your presentation. Professor Stenlik comes from University of Valparaiso, if I'm telling correct, from Chile. And his talk is on hybrid and self-adaptive spoke-based neural networks in health strategies. Milan, the floor is yours. You can start. Thank you.
Thank you very much for a nice introduction. And again, thanks for the invitation for this conference. I will be working on extension of the artificial intelligence to the hybrid and self-adaptive networks, where we need a specific activation functions, and I will explain some aspects important to the health strategies from the projects we have been having in the past 5 years. So for the outlines, I will be first learning we can take by aggregation, which will mean to find the proper dependent structure between the data monitored by wastewater monitoring for zoonotic diseases, which have been part of this conference. For instance, in the case of the SARS-CoV-2, and the same in infected or reported cases in the human population. So there are two streams of the data, how we can aggregate and learn from it. What is the correct approach here? The second health related topic will be adaptive and hybrid networks for improving the cancer diagnostics. We are still having a very bad false positives and false negatives in the discrimination between cancers and also on the diagnostics. How we can improve this? And the last will be the robust learning by aggregation of growth characteristic of bacteria, which relate also to one of the day's topics of this event, not only on the resistancy, but also to understand the range behaviors of the bacteria. We are having many bacteria, maybe more than stars in the universe, and we need to understand the proper I would say real and asymptotical properties of this growth. So example one, again said, is about the wastewater modeling. I have seen several talks addressing wastewater, and we have been also having a project in South America comparing the Peru and Chile. And there have been 4 papers which I would like to point out and discuss a little bit about the results on the next slides. So in the first paper, which is published in Stochastic Analysis and Applications, I will say that's not a fun to predict. And I like the question at the end of the round discussion from Publicum about the predictions for the future. Predictions are not working well. I need to say we have a lot of work to develop because when we, for instance, model the zoonotic diseases based on the reported cases of humans, almost all models, however complicated, have been losing their precision in 2 weeks, 1 week. There was such a issue discussed also in the scientific journals. You can see also this paper if you want to see more. So modeling and prediction needs a feature which we call adaptability or adaptivity of the network. So the network needs to be able to adapt to the process during the run. Otherwise, the features will not be predicted in a good precision after several time events. The second paper published in the Science and Total Environment is showing how we have been monitoring the surveillance of the SARS-CoV-2, rotavirus, norovirus, and human adenovirus in wastewaters as an epidemiological tools in practically Peru and Chile. Peruvian highlights, these are the upper parts of the Andes, which are still populated. So it's very up in comparison to the Europe, but they have a wastewater treatment plant there, and they want to see also the zoonotic viruses and relation to the what is happening in a human host population. And on the third paper is showing the reason why this kind of relations are not working well, because we need to understand which kind of relation between humans and virus we model, because there are delays and there are the regimes where the viruses are emigrating from the wastewaters or they're entering the wastewater. So there are some issues which have been published in chemometrics and intelligent laboratory systems. And there was a technology for effective monitoring by adaptive cluster sampling. This is not our development. This was used in Africa for at least a decade for modeling the HIV. And we can use the adaptive cluster sampling also in regional emergency driven plans for effective COVID management. This was again published in this journal. So I will go now to the more details to the wastewater. The project was between the Chile and Peru, running from 21 to 23. The name of the project was Spatial Temporal Evaluation of the Concentration of SARS-CoV-2 in Urban Wastewater Samples, and a relationship was happening in the population. That means practically we have been having a reported cases, administrative data from the region of Peru, the central zone of Peru, and comparing as a benchmark also what was happening in Chile. So that means practically several 1,000 miles difference in the space. Question, can we compare? Is this kind of method generalizable between the countries? If we are interested on selected vectors, practically SARS-CoV-2, rotavirus, norovirus, and genogroup of the genogroup type 2, and the human adenovirus in wastewaters. Well, that was not an easy thing, and we have been comparing this experiment also when monitoring 3 Central European countries, Czech Republic, Austria, and Slovakia, as a triangle. And we have seen there are a lot of challenging to model that, and there is not a easy forecast model. And we need adaptive networks. This is why I'm speaking about the hybrid adaptive networks today. And we have been publishing that paper, which I already have been having on a slide in stochastic analysis and applications. One of the problems we have here is that nevertheless, we are pretty much trained in modeling. Nobody defined the rules, how we can relate what is happening in a human population with the zoonotic, with the normal wastewater virus detection. So how can I measure it? Can I use the correlations? Well, we need to answer all of these questions which I have posted here, which have been bringing us to the learning. So first, correlations are related to linear dependencies, but there was a lot of non-linearity in the data, also in the wastewater data, and also in reported cases in Peru and Chile. Also the same for Austria, Czech Republic, and Slovakia. The second thing is that there have been always problems with getting correctly supported data because we cannot measure everywhere and all the time. So our measurements will be always discrete and there will be practically sparse on some places, which can be important for seeing the relations, especially based on a correlations type of measures. Range constraints and variability have been an issue for modeling. Which kind of model will be actually taking the variability? What is the noise and what is the data? Atypical values can be always visible in administrative samples. We can statistically address measurement errors, but not the systematic errors. And there have been asymmetric dependencies from water to population. What does this mean? What is happening, what is visible in the water, it is not exactly in the same time scale with the population and vice versa. So we need to know whether we want to address now population from the water or water from the population and how to measure these relationships. Just to show the example from the project in Peru, if we take one upper Peruvian position where we measured, which is called Huancayo, there have been COVID-19 SARS-CoV-19 cases measured in the residual water, 100 milliliter version of the maps. And we can see here the values of all these points are correlations. But which correlations are? The blue one is a Spearman, the violet is Winserized correlation, the green is Pearson, and the red is Jackknife. So if you take of these correlations, you can see all kind of relationships from increasing correlation to almost constant correlation. So you need to really, this is labyrinthic, and you can of course take all other types of dependencies to see that practically there's not an easy way how to get data from the how to learn from the data, the relationship between what's happening in humans and what's happening in the wastewaters. Well, the answer from this is that we need to build up the hybrid systems. What means the hybrid systems? One of the possible hybrid system is that you need to have modules where you have a network, for instance, artificial neural network or any kind of fuzzy network, which is defining you the levels of the relationship. You need to put a correct activation functions on the points. This is the technical part of my talk. There is an example too. Example 2 is a project which we are practically closing this year, and this is related to the simulation of real bacteria adaptabilities. We don't have this kind of studies because you can take physical parameters, you can take chemical parameters, you can take other biological, microbiological parameters, change the range, and watch about the adaptability of the simple one-cellular bacteria or different bacteria. It can be E. coli, it can be the Cobetia, which we have been having in our project. There is 3 papers which can be of interest for the people who like to see the resistance with respect to the physical and chemical conditions and reactions on the growth properties and also genetical changes of the adaptability practically. We can call it in these simple organisms. So there was a paper which is published practically now in the PLOS one, which was also partially presented in ICRA in the Patras organized very good conference, where we have realized that growth and biofilms, which is practically the shelter for the Cobetia and inadequate conditions. Both are very dynamically behaving, and they are depending on the temperatures, acidity, and other chemical and physical parameters. And we can model these, but we need adaptive hybrid networks because there is a beautiful adaptability. The bacteria doesn't have any brain, so we can think about even our constructions of artificial intelligence sometimes trying to mimic the brain. Maybe this is too far from what is really adaptive, because the bacteria doesn't have a brain. It is one cellular organism which is completely adaptive and can really react in a way we can learn from it. The effects on the pH, like acidity, have been published in Frontiers of Marine Science also this year. And there is a lot of development on comparisons, especially when you have heteroscedasticity measured on the variances, on the growth, and of the creation of biofilms. So again, we see that adaptability is one of the things we need for the systems, whether they will be artificial or there will be semi-artificial. There can be also non-human intelligence based on animal and bacterial behaviors. We don't need to think only about artificial intelligence. There is a lot of intelligence in the nature we can learn from, like simple bacteria, like resistance, like adaptability, like changes. Here is one of the pictures of the project. And again, if I want to model, I can try to model, I can try to predict, I can try to forecast, but again, it will not work if my system will not be adaptive. So I can have a system which will crash. I can have a dead gradient problem in neural networks because the gradient simply will vanish. But there's also generating us the question, what are the transfer functions which are not exactly non-adaptive and they can adapt to the changes during the run? This was a project you can see here. One of the most important thing is that you need to have a very good luck in tuning of the networks, and you need to be very critical on the inputs and outputs, which kind of activations you will put in the first layers. Then when you have a deep neural network, then the hidden layers, there should be some adaptive functions. Otherwise, you can easily lose, and there will be a big discrepancy like we have on the left hand, on the grow and on the real grow and simulated system, and a very good fit if you are adaptive in that moment on the right hand panel. And you can see here the several grows based on the temperature ranges we can have on the bacteria scale in the ocean. Well, which is telling us again, we will need a lot of work, not only on the IT position, but we need to come back and understand better the geometry of the learning. And that was a Nobel Prize for decoding of brain scenes of place, which have been actually generated from high-dimensional approach on the topological data analysis. So I'm here telling that yes, several things will need interactions, adaptability, but we also need the deep results in this kind of classical disciplines like mathematics, statistics, and topology, deep results. One of the things I would say, what is the adaptability for understanding of the general public? It also means that you have some memory on the recurrence event. Recurrency of the event, that's a pretty dynamical systems like thing, but it will mean that if I will take some methanogenic bacteria and I'm measuring the produced methane over the 1 year, then you are watching on the production of the bacteria, and you can make a trace, and you are watching whether the pictures which you create from this tracing are repeating. And what is the percolation number, which means that I can move over the black boxes from the top to the bottom. what is the percolation number of these kind of pictures. And this can build up recurrency patterns, and it also relates to the adaptability. The adaptive neural networks should be able to learn the recurrency patterns over the time, and they need to put it in the brain of the neural network. If I'm modeling the neural network, it can be hybrid system, which is mixing more models. Like one of the things is aggregation of pictures, objects, images, and probabilities by the simple black box machine. Multi-criteria ranking, inputs are coming, output is going out. This is related to all of the systems we can use for health, including the diagnostical systems, wastewater surveillance systems, monitoring of the blood pressure, anything, and that we need to merge several conflicting preferences. Or conflict is always present in this kind of mixing in a single decision systems. And one of the things I would like to say as an example to motivate and finalize the introduction of the SPOCU functions, that the most simple models which is used in statistical learning, in machine learning, is the neural network. Again, say inputs are typically multiple, outputs can be also multiple, but let's have just a single output layer, and there are several hidden layers here. Now, which kind of functions will I put on the layers? On the input and output, I can put practically any sigmoid like non-adaptive functions, but if I don't put on any hidden layer adaptive function, my network will not be adaptive. There's a message. That is having a link to the developments in aggregation theories, which goes up to the very crisp level on the aggregation function stories. But aggregation is each arithmetic mean, copula's products, these are aggregation operators. And we can aggregate the words, images, sentences, symbols, etc. There have been a theoretical discussion by myself in 2016 on that. Practically, you can whatever object you aggregate. Why we need to aggregate and why we need to learn the recurrent patterns to avoid the tragedies in the systems like self-driving cars. Tesla experiment US Highway 101 in Mountain View, California, 2018. The car has been overloaded in the memory, not able to see the barrier, and the driver who was been sleeping during that period has died after the crash to the barrier. Why the barrier was not seen? Simply the memory was not enough to big to have the barrier there. And if we don't do this thing practically by learning the recurrency events in a complex data flow, that will be happening in whatever system we will have. That is not adaptive system. This is not an adaptive system because it is not able to adapt to the increase of the complexity on the recurrence pattern level. So this is one of the things we can solve here.
Milan, have 5 minutes.
Thank you very much. It will be now coming to the closure. The cancer discrimination is the last example. Well, that's a thing we need to really work on. Why? Even in the simplest cases and the most protocolized 4 protocol type cancer like the breast cancer by women, we are still on the beginning of finding how to decrease the false positivity. That's impossible to solve after 100 years of protocol. So come on, let's work on it. So there is a cancer that here histopathologies on the right hand side. Left hand side is a mastopathy, which is practically style of the tissue lesions which are not carcinogenic, and typically they are linked to the diabetes and other type of comorbidities of the women's, many times premenopausal. So we have make a first step in 2015 to understand why the false positivity happens here, and we learn that even whatever the machines we will use, we are not able to beat 80%. Oh, well, come on, what's happening? Well, why we are still developing such a primitive systems and why we don't go to the deep diving to the adaptive systems. That was the question. And we have been working on it, and we have realized the same thing as we have seen today on the recurrent patterns by the bacteria, that if I will make up, this is a German version of the oncology, so you make a CCD camera, histopathology on the left-hand side is from the cancer, and on the right is mastopathy, and you can see the simple fractal dimension difference, much higher fractality on the left-hand side. There's a lot of genetic variability, which we cannot even explain by stochasticity. There have been a paper on the frontiers on oncology that even in a practically single genome, means you will have a correct compact supported distribution of the fractal characteristics. So what we did, we have been learning on this, the adaptive machine that will be at least so intelligent as the Querma's interaction processes on the stochastic geometry, which is related to the creation of the cancer tissues. Not easy job. Dysregulation using modeling epigenetic, which is a modern topic. This is a Nature paper from something like 2019, 2013, the first by Tim Feinberg. And again, we need activation functions which are able to learn this kind of geometries. Improving sigmoid. I had been having a project in Austria and Lean Institute of Technology about this. Neural network should be used. And finally, we ended with this kind of SPOCU transfer functions. So everybody want to implement can see, well, one of the adaptive functions which exist and have been developed to fight mammary cancer false positivity and increasing the discrimination between mammary cancer and mastopathy. It is practically, these are two technical slides. Having 4 hyperparameters, I need 4 hyperparameters to mimic the adaptivity of this kind of processes, which are very natural, because cancer growth is a natural issue. And I can build up the SPOCU to be the smooth, so I can use the gradient algorithms and to have all properties like cell normalization, identity near zero, monotone derivatives. We don't have, but we have monotonicity of the function on the most of the profiles. And checked on this kind of network can be checked on the comparison with the relevant cell, showing that this kind of function is adaptive and can model reported cases of SARS-CoV in triangle Austria, Slovakia, Czech Republic. It is able to adapt to the bacteria Cometia growth modeling. It is able to increase the cancer discrimination quality, improving the metrics in the Wisconsin diagnostic breast cancer data set. And we also compared with the CELU and the RELU on a large Ministry of National Institute of Standard Technology dataset of the cancer and outperforming that. So that I'm saying that adaptivity is a very important topic we need to develop. Otherwise, we can say technically that we outperform over the introduced activation functions on the deep levels of the neural networks, and we can also build up the search algorithms which guarantee the convergence on the hyperparameters on the high dimensional settings. In a summary, I'm again claiming that we need adaptivity. So what means adaptivity for us? It can help us to adapt to this complex period also on the health issues and climate challenges we have here. But we need also adaptivity from the perspective of the techniques we are using. Also ourselves, but also the techniques. So I'm acknowledging several projects here. And Eucharisto, thank you for your attention. I hope I can give some feedback.
Okay, let's thank the speaker. Any question, comments to the speaker? From online, do you have any? No. If not, Milan, may I ask something?
Of course. Shall I stop screen or shall
Oh,
you?
no, it's as a general question. The Spoku-based neural networks are available on some software, or how can
Yes.
use it, try them?
Very good question, Polychronis. It is, we have made fixed parameters implementation to the PyTorch. There is a Keras link stuff, and we want to develop now the algorithm which will be adaptively changing the parameters because there are 4 hyperparameters. But there are fixed versions of the parameter setups are available, and we are making a lot of experiments of that, of course.
Okay, and you can share them or not yet?
Sorry?
You can share them or not yet?
In this moment, you can use fixed parameter implementation and I'm available to send the links to the codes. But in this, what we are developing is adaptive strategy for the parameters based on the depth of search, which is still not finished. But on the given parameters which are working well, for instance, for these examples I have been showing, we have it implemented.
Okay. Okay, thank you. Any other question? No? Okay, thank you, Milan.
Thank you very much.
And let's move to the next speaker, Adrian Egli, if I'm pronouncing it correctly. I'm sorry if not. He's here. Okay. Can you hear us?
I can hear you very well.
Okay,
can you hear me?
Okay, yes. You can see your presentation. He's going to present a talk on epidemiological modeling using AI. It's a different title from the program, but it's okay. So the floor is yours. You can start.
Thank you very much. Thank you very much for the opportunity. What I will try to do is give you, first of all, a brief introduction into AI, but I realize that you have heard from the previous speaker about it. And then I will try to use how this technology can be used in medical microbiology and here specifically epidemiology. I'm very sorry if I use here now a different title. I apologize for that. So the maybe line out of my talk is as follows. First of all, why AI? Why now? Then I will give you a couple of examples on use cases basically, and then we will also talk about critical aspects and challenges using artificial intelligence. But first of all, we have to realize that we are in a extremely rapidly advancing time in terms of technological advancements. You can see here computer technology evolution over the past 30 years. The Intel 486, that was my personal first computer I had. And nowadays you have extremely powerful graphical processor units, which are in terms of calculation capacity about 250 million times faster. That's unbelievable if you think about it. And the consequence of this is, if you look at this time scale here on the right, you can see here different machine learning achievements. So for example, handwriting of digital recognition systems. And then here you have large language models now and more sophisticated artificial intelligence systems. And you see here the training they actually need, the training compute. So how many calculations you need to train. And you can see that this is clearly in an exponential period here. And some of the most advanced ones have actually more calculations than that are stars in the universe. There's an estimate that there are 10 to the power of 25 stars in the universe, and this graph is already outdated. So it's an absolutely amazing time we live in, and there are certain consequences of this, obviously, that we can use machine learning and artificial intelligence. Now, artificial intelligence is very good in two things. The first one is categorization. You can see here an example of muffins and chihuahuas, and they look very much alike. And obviously, you don't want to bite into a chihuahua, but you only want to bite into the muffin. So what can you use? You can use artificial intelligence to kind of separate them and to build categories. And this is something which we always do in medicine. We classify, we categorize gram-negative, gram-positive, antibiotic drug resistance, antibiotic susceptibility, and so on. And black box assays, like neural networks, for example, help us to do that. There's a clear trend to open this black box and make it more explainable. If you look into the literature, this is the so-called explainable AI. So that's the first use case, is categorization. The second use case is access of information. And this is not the same as knowledge. We have to be aware of that. But it's really remarkable how large language models can process huge amount of information, not just text, but also they became multimodal. So they can also look into image, video data, speech, and so on. And you see here on the y-axis, again, a time scale. On the x-axis, on the y-axis, you have here the percentage of correct answers in the board exam in the US for medical students. So MedPalm-2 is a large language model from Google, and it had 86.5% correct 2 years ago. Now, if you extrapolate this curve, you can imagine that this is now much, much better. So indeed, these large language models easily pass the board exams for medical students. Now, the point is that, you know, medical studies are obviously very broad in terms of knowledge, but you, the audience, and myself, we are experts in certain fields like public health, medical microbiology, epidemiology, and there's obviously less training data around for the machine to learn. So most likely there is data scarcity, and because medicine is per se a very heterogeneous topic, it could be that in our fields and in specialized fields, the performance is not yet as good. What we have to be aware of is that chatbots and large language models, they rapidly evolve. Obviously, ChatGPT 3.5 had huge issues with hallucinations because it was trained with the internet to a certain time point, and then it was a static database where the chatbot did predictions of the next word within this data space. But this has radically changed. If you now go for ChatGPT 5.1, it's agent-based. So what this means is you generate a prompt, the prompt is added to the chatbot, and the chatbot does the following. It accesses real-time new information. So it can access a database. This could be PubMed, for example. It could be the European Nucleotide Archive. It can be the internet, anything you like. If you have an internal large language model in your institute, for example, or hospital, it could access, for example, SOPs or PhD theses of your students and so on. And then it analyzes the text and it generates an output. So it's real time. And this drastically reduced the danger of hallucination. Now, you can use this for epidemiological purposes, and I give you here an example. I generated here a prompt where I said that I'm a medical microbiologist, and it should actually access public health websites of at least 10 European countries, and it should generate the incidence rates of influenza cases, because we now come into the season of influenza, from the beginning of the year over a series of weeks. And it should visualize the output in a table. Then it asked me a couple of questions back, and I gave some answers. Yes, please just focus on some countries. Keep it simple, I said. Like, I don't want fancy statistics about it. I just want a simple table. And what was the output I got? Is the following. You can see here every week over these 8 weeks. You see here the countries, and you can see here the incidence numbers the chatbot found on official websites. So here you can see some links where it actually looked up the information. And when you click on these links, you can verify that these numbers are correct. And so I think that's a quite remarkable and helpful tool if you want to access information, because you can access information across different language barriers. You can really generate it quite accurate. And for example, a chatbot, you could program an agent now who does this on a regular level. So you get very customized information within a short time period. So now I would like to make it a bit interactive, and I would kindly ask you to scan the QR code and help me answer this question. Have you ever used a large language model to code anything? I mean, you can answer it now for epidemiology, but you could also go and answer it for an epidemiological analysis or any type of analysis. You can scan it. Let's wait for a moment. And with coding, I mean like writing R code or Python code. So let's wait until we have a few more people. Okay, 7, 8. So I would say 2/3 have used it for coding. And the obvious question is, why did you use this for coding? And I think because it's very good in coding. And I give you an example for this as well. What I did is I looked for a training data set for students. And this was an epidemiological data set of John Snow's outbreak of cholera in London. You probably are all familiar with this because this was one of the first epidemiological case studies in 1855. And I basically uploaded the Excel sheets into ChatGPT, and I asked, help me understand the data. This is a historical data set from a potential outbreak. Can you analyze the data for me? And what it did, it said, yes, I can. And then, very interestingly, the chatbot itself decided to write some code. And it generated, for example, you can see here the code, it generated a map, a visualized map. So each red dot here is a cholera case, and there were some geographical information on distance to the water pumps in London. And you can see here this visualization which was generated. And I verified the code, and this is all fine, what it generated as sort of output. Then it also generated epicurves, so for example, on mortality and case rates, etc. So that was quite impressive, I have to say. Excuse me, that the chatbot generated this output. Okay, now chatbots are really good in coding. And this was quantified. You see here a link where this was measured. You have here a timescale again, beginning of 2025. And probably a few of you are playing chess, and you're familiar with the ELO rating, which is a way to assess the skill set of a chess player. Now you can use a similar rating to assess the skills of a human coder. And this dashed line is where 90% of the human coders have a ELO score below 1,600. That's here. 90% of humans are here. And now you see all these chatbots here are way above, and we are here in the exponential code. And this is now the reason why nobody on the market is looking anymore for people who do software development, because at the end of the day, this chatbot can simply write the code for you. And this has an impact in medicine, because if you now have an outbreak and you need the software to analyze it, obviously a chatbot can help you to generate this data or generate the software, which helps you very rapidly. Okay. Now, Another interesting use case for AI is AI for modeling epidemics. And I want to give you here now a few concrete examples from a fantastic review article which was published in Nature beginning of this year. And the idea here is that foundation models are used, so transformer-based AI, for epidemic time series analysis. And transformer-based models are very data heavy. So the input data would be a case incidence data with a potential gap or missing point. You see here this example. You have here an epi curve over time again. You see that there is missing data indicated here by this gray bar. Then you have some outliers. And then you have the future where you do not know what's happening. Is this going down? Is this going up again? And so on. So that's future prediction. And the training data would be a transformer architecture trained on empirical and simulated epidemic data. So for example, if we take the influenza season now, you would go and take from your local city all the influenza cases of the past 10 years, and you could use this information to kind of simulate the dynamic of an influenza epidemic. So that's quite nice because it could help you to guide resource allocation and evaluate interventional effectiveness. The second use case is here. So that's graph neural networks and to analyze infectious disease spread. So the idea is that nodes represent locations and individuals. So you can see here a network, and every dot here is a node representing a case or an individual. And then edges represent the transmission pathways. So you can see, for example, I don't know, these would be two individuals, but it can also, this could be a food compartment, and that's maybe people attending the restaurant, or I don't know. I mean, you can be here really creative in looking into this, and the features then include disease cases, demographic information, and for example, in the case of a pandemic, you could also integrate healthcare capacity. And what the GNN does, it classifies those nodes. It can help you to community detect. It can help you to identify hidden transmission routes. So for example, you can say, oh, here is something unusual happening. Could this be a transmission route? And for public health, this clarifies to understand the spread dynamics of an infectious disease. The third use case is protein prediction. So predicting immune escape mutations using deep learning. So as you know, viruses adapt all the time. This is the reason why we need to vaccinate every year. And so what you can use is structural predictive tools like AlphaFold or ASMFold, which help you to translate the amino acid sequence into a 3D structure of the protein and to analyze, and this could be based, for example, on viral genomes. And then it identifies mutations disrupting antibody binding, so in a mutation which has a severe consequence because the vaccine antibody would no longer bind, and it indicates then potential vaccine resistance. This is obviously very, very important for public health actions. And in this case, for example, this could be the hemagglutinin of interferon of influenza. Okay, so these are three use cases, but what is out there in the literature in terms of epi modeling? So what AI-infused models for infectious diseases exist? And this is another very nice review article, also published beginning of this year in Nature Communication. And the authors did a marvelous job in summarizing different aspects. You can see here a so-called Sankey diagram where you see how the research is organized. So you have here the application area. So for example, you see here at the bottom, outbreak detection, or model parametrization and calibration, or you have infectious disease forecasting. Then you see the methodological framework, what kind of AI model is basically used or machine learning model is used. And then you have the integration approach. So for example, integrating the epidemiological knowledge into an AI model. Now, there are 9 use cases discussed in this review article, and I will just show you 3. And let's start with something you're most likely familiar with. So you can see here, so here the method basically is an AI model trained to predict an unknown epidemiological parameter. So you have here an epicurve, and you want to generate from the epicurve or you want to feed information into an SIR model. So susceptible, infected, resistant model, a classical epidemiological model. And there you have certain parameters which, for example, influence the transmission from one compartment to the next. And these variables you can obviously compute. So you can predict, for example, reproduction numbers of recurrent neural networks. And this really helps you to enhance your model. The second example is using AI to enhance observational data. That's the example I've shown you before, where you have missing data in your epicurve, and the machine learning model can help you to fill the gaps and thereby make this model also much more reliable because you have less gaps in the data set. The third one is using clustering techniques to group regions with disease transmission characteristics. So for example, you have region attributes, let's say different cities in Greece or in Switzerland. These would be cities, for example. There are region attributes here to these cities because, for example, the transmission dynamic in these cities is different because the population density is different, the behavior is different, and so on. And so you can use this then in individual SIR models, regional attributes into an SIR model, and other regional attributes. And then you generate specific projections based on these regional attributes for an epicurve. It's also very, very an interesting use case. So there are lots of publications on different approaches, lots of opportunities and models, but there is hardly any real-world prospective usage of these models. And I think that's a huge downfall. All the publications which I have found in this area for this presentation here are retrospective. I've not seen a public health authority which would actually use a prospective model. So this slide I will skip for the interest of time. I want to focus a bit on the challenges, the critical aspects of data bias. We live in a world where we have more and more data available, especially in diagnostics and in medical microbiology. As an example, in 2030, we will reach 612 zettabyte of information. This is absolutely ridiculous in terms of the number, but you can see that this data is completely biased where it comes from. So lots of data from the US, lots of data is stored in China or generated in China, but it's not likely leaving China. And there are huge gaps in where the data is coming from. And you can see this here. And this is just an analogy here. This is street map contribution. So if you go on Google Street Map, you see here the dark blue areas. This is where street map is popular. And the white areas, there is basically no street map data. And you can see the bubble size indicates the population density and the population centers. So there's a bias in where is data coming from. And that's important when we train machine learning models. Because if you train something in Europe and then you have a patient from, let's say, India, you cannot be sure if this actually works. And during the pandemic, this became obvious. So you see here, the more dark blue it is, the more sequencing was done. And these blue areas here, this is Europe, European countries. And many other countries have very scarce, sequencing efforts. Now, collecting data.
Professor, you have 5 minutes.
Yes, thank you. Collecting data in a structured way is very important, and there are different incentives to do that. We have one in Switzerland, the Swiss Pathogen Surveillance Platform. There's an Australian version, the OSTracker. There are certainly also, incentives in Greece where, for example, sequencing data and surveillance data is collected. And I think that's a current trend in Europe, that we start to collect the data, that we analyze the data in a harmonized way, and that you then share the data with either public data repositories of federal offices. And the same, so in Switzerland happens, this is Australia, but there are many, many other examples. And I think it's very, very important that we have high quality data, which is well structured. Okay, what were my conclusions for this talk? The age of AI is coming, also for public health. Get prepared and make it a personal priority. You see it's in an exponential phase. So the role of the expert will be challenged by some of these machine learning models. Maybe not tomorrow, maybe not in a year, but in 10 years, I can guarantee you our job will look very, very different. And we need to prepare for that. The challenges are heterogeneity of the data, data gaps, retrospective validation, single center studies. Nothing is externally validated at the moment, but I'm pretty sure that these challenges will be tackled over the next years. The development is extremely fast. There's a huge potential, especially hospital administrators. They believe that, you know, everything can be solved with AI. That's obviously not true. We need critical evaluation, but this will also come in medicine. And in areas where you have high data standardization, especially in the diagnostic lab, AI-based diagnostics can really be deployed and used. So we need a careful evaluation of AI-based tools, no matter if this is in epidemiology or in diagnostics. It's needed, but I'm pretty sure this will come. And I'm very happy now to answer questions. You can follow me on LinkedIn or drop me an email, or I'm happy to answer questions now.
Okay, thank you. Any question from the audience? Yes.
Hello. Thank you for this amazing talk. I really enjoyed that. I'm Professor Apostolos Vandarakis from Medical School, University of Patras. I was wondering, you said something about we don't have real data for use of AI in epidemics. The most of the articles are with retrospective data. Is that correct?
Yes.
Yeah, I understand it correct. Yeah. So
yes. What? Yeah.
Sorry. Tell.
Go ahead. Yes.
I'm just wondering how close you believe that we have the ability. We will have the ability to use in real situations, in real time situations, AI. I mean, we have an epidemic, and we would like to have the model on these epidemics. I would like to have your opinion on that.
Yes. So I think, first of all, I mean, a very important point, and I think with AI is the trust we have into these algorithms. I mean, at least my impression was that during the pandemic, we had a lot of newspaper article where epidemiologists were asked, can you predict the future? You know, and they had certain models. And then whenever an epidemiologist was wrong, this was more often in the newspaper than when the epidemiologist was right. So I think that's a problem. And due to that, we need a careful evaluation. And I would actually do this, you know, in a real world setting now, when we have now epidemics, let's say influenza or others, that you actually try out models and you really see which one is the best model, but you try to also inform the public health authorities with this information. And then they can decide and maybe talk to other experts. I would anyways not solely trust an AI. I mean, you also need to think and, you know, reflect the output of the AI. But then I'm pretty sure that this should be done. And you know, the thing is, the next pandemic is coming. At a certain point, we will face another pandemic. Likely, this is going to be a bird flu virus, as it looks in Europe. I mean, H5N1 is spreading everywhere. And as soon as there's an adaptation to human hosts, we will have a pandemic situation again. And then it would be great to have such models available. And I think a lot has happened between the pandemic and now already in this AI field. Yeah.
Okay, thank you. Yes, have another question.
Hey, Uli Klumper from TU Dresden in Germany. So AI is big, and I believe that even if right now we don't see it implemented, it will soon be in there. So I totally agree with you on there. It also is, if we use it in health, obviously a security topic of like great defense importance. So if we want to actually implement it, do we need our own European large language models to run this stuff? Or is it possible to implement the ones that, well, that are most commercially available right now?
Yeah, the thing is, the Americans have the most advanced models. And I think it's quite unrealistic that Europe will catch up in terms of the performance of some of these large language models, simply due to the fact if you take the most powerful European developed large language models, they really fall behind simply due to the fact that for example, Grok, from Elon Musk, or if you take ChatGPT, there were billions invested into this. So they are extremely powerful. Now, what's the way around it is smart design. So the Chinese have shown that with DeepSeek, so that you can generate a large language model which uses less compute power. That's a way around it. And the other thing is that you use pre-trained models which are specialized on a certain task, and then you can run them much more cost efficient. And I think what the future will bring is that most institutions, hospitals, universities, maybe even on your own laptop, you can use then internal large language models which are suitable for certain tasks. So if you take an open source model like Llama, Llama 2 kind of sucks because it's not really good, but I mean, you can run this on your own laptop. So that's not the problem. And I think also these models will become much more efficient over time. I hope I answered the question. I think, and then be careful what you share. You should never upload confidential information, obviously. Yeah, well, that's an obvious one.
Okay, thank you. Any other question? If no, let's thank all the speakers once again.
Thank you very much.
Thank you.
Bye-bye.
Thank you. Bye. Now we go to a coffee break, right? Yes, okay.
Yes, yes. Let's go for a coffee break.
Good afternoon, everyone. I hope you enjoyed the coffee break. And now we are back with a last session. And I'll invite Ioannis Kyuvrekis, Assistant Professor at the Department of Public and One Health in the University of Thessaly, Greece, to organize the workshop with title Artificial Intelligence in Practice, Selecting and Evaluating the Optimal Model. Professor Kyuvrekis.
Thank you. The idea was to have an interactive workshop with the audience. So I want help from the audience. And I want you to answer a question. For you, what is machine learning? I know that Athanasia, for example, used for his PhD some ML algorithms, random forms, etc. So I want you to answer what machine learning is for you. What do you believe that is machine learning? The classification, the regression problems, artificial intelligence. Can you define with simple words what is? Yes, try. Yes. Not only Athanasia.
I will make the start. In simple words, I can say that is a way for, I will call it a model for a computer, maybe, to have data, inputs and outputs. So the model must know what we want to estimate, to calculate, what is the result. It can study the possible relationships between inputs and outputs. It's being trained, and then by itself, it can make predictions, estimations. So it's a way for computer to make its own predictions.
Okay.
To have a brain, possibly by itself.
Brain?
Yeah, metaphorically speaking.
Okay. Someone else?
So I'm not an expert on machine learning, but from what I have perceived in believing that machine learning is, I believe that it is a way of us to construct a structured form of analyzing pattern and data, but the algorithm does this for you. You don't have to give them a structure to do that. You just train them with data, you input data, and they give you an output.
Okay, thank you. Someone else?
So I think for me, as completely, I always say I don't know anything about statistics as a scientist, apart from the stuff I can do in Excel. So for me, it's a way of looking for patterns without having to think about all of the ways that my input variables are interacting in before. So I don't need to do any statistical corrections myself because it is automatically done by looking at certain things.
Okay.
Yeah.
Okay.
And if I can say something as well.
Of course.
Well, from a sociological perspective, because I think that for me, what I would like it to be at least, or what it could be, is a tool that makes our work, our life, and our discussions easier in relation to what Uli was referring. For example, things that we could avoid. We can avoid. What strikes me, though, as a problematic thing, if I may, is, and maybe it's a point of discussion in the workshop later on, but I'm just mentioning it now, is that it seems to me that the algorithm seems to, get to know us and seems to always giving us answers according to what we want to hear. So it
Really?
learns us. It learns us so much that no matter how good are the questions, it will eventually, give us answers on the basis of what it knows that we like.
Okay. First of all, you refer to algorithm or algorithms like an identity, like a person. And secondly, you said a model, that in machine learning we have models. Can you define what a model is? Not next top model, but machine learning models. Or what can be a model in machine learning problems? Okay.
So my only connection with machine learning is asking ChatGPT or Gemini to write for me an email. But I can understand that it is a system that can recognize some patterns and build a data system to make a decision or a conclusion for some.
Okay. Now, a model, someone else about the model, what a model is, or what can be a model?
For me, as I have learned or studied, I see machine learning as another aspect of a calculation, a calculator. If I do 1 + 1 in a calculator, it would be 2. So in my perception, the models are most difficult mathematical
objects.
Equations, or my challenge to understand is the difference with the epidemiological models and the statistical aspect of this kind of situation.
Very nice. Do we know what epidemiological model is? A system of differential equations. Machine learning models can be very simple mathematical objects, like a line in the plane, a line. A line can be, and it is, a machine learning model. Why? Because we can embed our data in vector space, in a plane, in two-dimensional space. And the one goal is to find a model in order to separate our data, the points. The data are points, and we want to separate them. Cancer, no cancer. Dogs, cats, 0, 1. The supervised learning, it's not something supernatural. No. It's a method to estimate a function. Just a function. The f function. Okay. You don't know the formula of the function, because if you know the formula, you know the rule which assigns an object from X, the domain, to an object in Y, the codomain. Okay. So classification and regression problems where we use decision tree, random forest, neural networks, deep learning methods, XGB boost, current neural networks, etc., 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 can formalize in a very simple question, which is the function f. In machine learning, we try to find the function f using a new function, the h, the predictor. So, the first thing that we do is to define in formal framework what is learning. Because we talk about machine learning and the algorithms, blah, 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 but what is learning? Okay. The first definition is the domain X, the set of objects to be labeled. For example, set of features, images, spectra. All of these can be the elements of X. And we have the label set. Are you a cat, a dog, an image of cancer, image of digital numbers. Are you 0? Are you 1? What label are you? Okay. And in real-world problems, we don't know all the space. We just take a sample, the dataset, the initial dataset. It's a sample from an infinite space. Okay. And we know that there exists a true labeling function, such that all labels in the sample satisfy y is equal to an image of xy. It means that, in simple words, for example, if I have to classify the digital numbers from 0 to 9, I have some images. Images consist of pixels, and I know F knows all the possible combinations in pixels, and which combinations gives 0, 1, 2, 3, 4, 5, 6, 7, 8, and 9. Okay. I don't know the F. The goal is to find a function H, a predictor, a classifier, a regressor, which simulates the F. Okay. Now, and the learning algorithm outputs a hypothesis, the function H, the model. Here, the model is just a function. Okay, function H. Now, how we measure our performance? We define the error. Okay, here is the true error. The true error is the measure, the probability where of the set where the H has different values of F. Is the set where we have done not correct prediction. Okay? Okay. Now, and our goal is to minimize this error. But we have a simple problem here. And I want your help again. Can you imagine which the problem is here? The F is the God, is someone who knows everything. I don't know everything. I must find a way to simulate this. That's why I construct using random forest, XGB boost, several machine learning methodologies, a function, the age. Okay, and I said that my goal is to minimize this error, but there is an issue here. Yes.
I think to me, the problem with those things has always been, how do I know what is a local minimum from an absolute minimum?
Okay. Very interesting. Okay. You mean that?
So, I mean, it looks like if I improve the model slightly in one direction or in another direction, it gets worse outcomes. So it looks like I have found the optimal solution. But maybe if I move to a completely other set of the formula H, then it might be that that formula H is better than the original one that I used, even if it looks like if I improve it only slightly, it gets worse.
Okay, and here, how can translate your opinion in our framework? Because we don't have local minimum, maximum.
Well, basically, I don't know how LDF minimized looks like outside of F. So I know with F it is 0. But if I say I cannot create F, which is rather likely, but I can just create an approximation.
Okay.
What is the smallest LDFH that I can actually create?
Okay.
And should I be happy with one that is at 0.1 or one that is at 0.05?
Another opinion? No one. The problem here is that we don't know the H or the F to find the probability, the measure. That's why we use the sample, our data set. Here, this measure, this metric, this error is calculated in the whole set of our data. Okay, but this is not the case in our problem because we have only a finite data set. Okay, so we don't know, we cannot know the true error. That's why we use some other metrics like empirical risk, for example. Okay, and this definition is very natural. It says that this risk is equal to the ratio of your success. Okay, and it's very natural. Can you understand why? Do you have any? Very quiet audience. As I said to my students, we don't make things to make us to make our life difficult. We make and define things to make our lives more easy. Here's a very natural definition of an error. Okay. And this kind of error can yield us to several issues in ML problems. The first and the most famous word in every talk of ML is overfitting. Okay. Do you know what overfitting is? Yes.
From what I've understood is we have the model and then we trained it with our data set, but it somehow made the model extremely accurate for the data set. But it's very accurate for the data set, but when it's almost only accurate for the data set. When it's the real life, it doesn't really work anymore because it's the only thing it knows is the data set. So it knows, okay, like this is, it overcorrected itself.
Overcorrected, okay, yes.
Something like that, I think, but I'm not sure. It's just, it's very, again, it's very accurate for the training, but it's not really accurate for anything else.
Okay, and the reason is? The reason?
I actually don't know.
Too complex. Which, the model or the data? Okay. Too many things. Okay. Overfitting has two branches. The first is the selection of the error. Here, we can select a metric of accuracy, which can give us false conclusions. For example, in our example, imagine that we have two squares with length 2 and 1. The little square has only blue points, and the other, the difference is red. Okay, and we try to find a way to estimate if you are blue or red. Okay, using the empirical risk. And we have a genius in our group who proposed this kind of predictor. Okay, this predictor says something very simple. If you are one of these blue points, you will have the correct label. Otherwise, I will put you 0 as a value. Okay. Now, in our training data set, this error has value equal to 0. Our accuracy is 100%. Okay, but the real error, can you imagine the value of the real error? No. It's quite simple, I believe. No. Not 1. 1, no. 50%. It's 50%. The true error for this example is 50%. Okay. But I have selected, a predictor and an metric which has drive us to false results. This is one branch of overfitting. The second, and the most famous, that we have a complicated model that can fit only to our data, is something like that. Imagine that we have the green line. The green line is a very common and famous function, sine x. Okay. We know sine x or cos x. Okay, I believe, yes. Okay. And we take some points, the blue one. Okay, and we add some noise also. The first idea is to make a model, a very simple model, just a line. Okay, a polynomial with degree 0. Okay, and we can use some not some simple mathematics, and we can interpolate in the blue points several polynomials. Okay. The last one with 9 degree is perfect, is a perfect model for our data set. Why? Yes. Exactly. Okay. And I'm happy with that. Exactly. In the previous model with just 3 degree, I'm very happy also, but I haven't 100% of accuracy. Okay. Now, why the last example give us an overfitting issue? You said almost, yes.
Okay, it fits the model, but the data we have, sorry, but with a very wrong way.
Okay.
With a way that is outside of reality, but within our own reality, it fits.
And how we can justify that?
It tries to cheat.
We try?
We try to cheat. The model tries to cheat.
Okay.
Or it's going to invent the perfect function for the particular numbers.
Mm.
Not such happy.
Okay. Let me put with another words. We have the problem to find a model in order to estimate the real values of blue points. Okay. And I have given 4 examples. In the last example, I have 0 mistakes. Okay. Why this is problem?
Probably because of the degree of relations that the model assumes doesn't fit the amount of data points that there are. So that was what I was earlier referring to as a too complex model.
Okay. No. I believe no. It is not an answer. I believe.
Okay.
Sorry, can I say something?
Yes, of course.
But if you take another sample from the same population,
yes,
then your model will not work properly because the other points of your model are outside from the real situation.
Okay, I agree. And which is the solution here?
To take another sample and another sample, and then
another sample, and
then mix together all of the samples results. I don't know.
One solution is to use, sample, another sample, another sample, another sample, and to test a lot of different models. If we have only one set, not probably, the last picture is an excellent model. Okay, the problem arises when we use unseen data. Okay, so in order to avoid overfitting or to avoid issues like overfitting, we use we want to use a very common methodology, and here is a problem in projects where involved people from other fields and not from mathematics or computer science. That this methodology is very restrict, and in a lot of papers and research papers, The research teams, they don't use with the appropriate way. Okay, so the idea is to use a lot of different data set, but also a lot of different models. Each machine learning model has inside some parameters, high parameters. So we have a problem to estimate the true function, but also inside the procedure of model selection, we have the problem to find the most appropriate combination of high parameters, the more appropriate model, not just in one dataset, but in a set of datasets. Okay, that's why we must split our data to 80, 20%, etc., a lot of times. Okay, in order to avoid this, in order to avoid this scheme. Okay, we want the pattern in the red and the blue lines to be the same. So the main idea is cross-validation, is the term, is to try to find the optimal model. Okay, and we must use a criteria, okay, to find the appropriate model. Do you have any idea which is the more appropriate way to choose our model? I have a lot of data set, the training and the test. I have a lot of models, mathematical models. Okay. I have the ingredation. And I find some errors for each model and for each data set. Okay. And the question is how I will reject the majority of the models and I will pick up the optimal. You make what?
I can make tests by
tests.
Yes, by using a specific data set with a couple of models, and to see and evaluate the results through metrics.
Which metric? I have chosen a metric. Okay, accuracy, recall, precision, something. F1 score, an abstract metric. Okay, but how I will choose the optimal model? Imagine that each model has been tested in 50 different pairs of training and test sets. How you can use select the optimal model?
The one who gives best results regarding the metrics.
What do you mean with the best results? What do you mean with the best results?
High accuracy.
High accuracy.
High prediction. A good F1 score.
Imagine I have only two models, neural networks and random forest. Okay. And I have one just combination of hyperparameters for each model. And I test these two models in the same 50 different pairs of training and test dataset. Each pair of training and test dataset give to the models a performance. Okay, so for each model, I have 50 performance. How can I select the optimal?
Some models correspond better to specific data sets, depending on the nature of the data and the relationships between them.
Okay. I will try a different question. Imagine that you must go to a student competition. Okay. Like a mathematical Olympiad or something like that. Okay. Which student or colleague you will choose and why? It's not a AI question. How will choose? Yes.
The highest GPA.
Which is?
The mean.
The mean,
Yeah.
exactly. No? Sorry, yes.
I would definitely pick the one that has the highest chance of winning it. So the one where the outcome is optimal for the highest amount of cases. Because I want the gold medal, I don't want the silver or bronze.
Okay, but I believe you said the same thing.
He said best average performance, I says most likelihood of peak performance.
Okay, thank you very much for your answers, because we have this issue. Yes, the most common answer is to choose the optimal model based on the average of the performance. But as, I don't know your name, Uli. As Uli said, maybe I don't want the average. Maybe I want something more higher. So it's not very common to choose based on the distribution, because this box plot, okay, box plot is not the distribution, but is a way to see how your model, the performance of your model in the set of 50 datasets. Now, for example, we have KNN, support vector machine, linear regression, tree-based model, and deep learning. Okay, and the y-axis about accuracy. Which model you will select for the optimal, and why?
I think I'd pick the deep learning.
I
think I'll pick the deep learning. Seems to have very high values that I can bank on.
Okay.
And I don't mind if it goes once in a while really bad.
Okay, and why not support vector machine, the second one?
Because it never reaches to the real top.
Okay. And
I would choose the third one.
The third one.
Yeah.
The linear regression. Why?
Yeah, because its lower values are above 85 of accuracy, and I know I will have accuracy from 85 to 95.
It's more stable.
Yes.
The variation.
No variation. Yes.
Yes. Depends of the domain expert here, because all the means, almost all the means for support vector machine, linear regression, and deep learning, the means, the average, are almost the same. And when I say almost the same, I mean that I must use another tool from statistics in order to find what?
If they differ.
If
they differ.
Yes.
If they are different.
Okay, I will use hypothesis test in order to find if there is a difference between the means of the models. But depends on the domain expert which of the three models I will use for the optimal model. Because the linear regression has the smallest variation of variation. Okay. But the deep learning and the support for machine has more high performance in some data set. Okay. So I think that, and I will close with that, the most difficult thing that I have met in ML projects is not to find a code and data in order to give an output. No, for me, the most difficult problem and situation is the use of a sound and complete methodology in order to select the optimal model. Okay, if there will be a universal way, a universal language, something like that, in order to all the teams can respect these steps, probably we'll have we'll have more accurate results. Okay, and this is, for me, it's very simple. We must just respect some steps. I know that we are the minority in the ML community, but we'll try. Okay. I hope that I will give you a different perspective what a ML project is. Thank you very much.
Thank you very much for your very interesting workshop. We learned things that we often don't see or neglect.
Yes. Is any question from the audience? No? Yes.
It's a very simple question. I was wondering why the probability was in the half, in the 50%, in the squares.
Because the area of the red square without the blue points are 2. If I remove the little square is 2, and the blue one is 1. Okay. But this is a very simple example how we can use, how we can select the not appropriate metric in our project. Okay. Also here, if I used another initial set, I will be able to understand that my first hypothesis is very wrong. Okay. That's why in the ML projects, we want to repeat again and again and again the procedure of model assessment. Okay.
Okay, thank you. Is there any other question? Yuli.
So for me, one of the things I always wonder is how much should I allow the model to know already? So how many of my own assumptions do I feed it before I let it learn? Or how many of the correlations that are, for example, that certain parameters are covariates, how much of it does it need to know before?
Before what?
Before I search for the best function. So for example, if I know that, let's say, pH and carbon dioxide in the water are correlating most of the times, does it need to know that I want those two to always be co-evaluated or not?
Okay, there's a misunderstanding. we don't try to find a correlation. Correlation, the problem of correlation is from another field, not from...
No, but I put in like both as individual data at the beginning, right? To get to results. Does it automatically condense those two to one predictive variable, or does it take it as two and then value it higher than certain other ones that don't correlate?
I have confused with your question. Sorry. Can you repeat or give me a more concrete example?
So for me, the question is, if I feed in data,
okay,
the different rankings of the different types of data, if I have a lot of them that are correlating with each other,
okay,
should I condense them before, or does it do it automatically, or should I put in what I know is scientifically true, that certain parameters are just basically defined through a certain relationship?
Okay, depends on the model and of the question. Yes, but this is the answer. Sorry, this is the answer. Yes, yes, of course, because...
Certainly depends on the problem that we have. From my point of view, I will try first let the data speak for themselves. Okay, that's the first approach. Because I'm a statistician, I believe in the data, I believe not in the data, not in your knowledge. Okay, that's true. I have trust in that. Data tells only the truth. Your opinion is personal opinion, personal knowledge. At the second step, I will go back to you and ask you about your expert opinion. If my results are aligned with your expert opinion, that's fine. End of close. We're both happy. That's okay. If it's not, we have to go through a discussion about what is missing. Is missing for my data not very good for the quality to extract the knowledge that is out there, or your knowledge is misleading. One of us has all the wrong data or the wrong knowledge. Then we're going to discuss. If your knowledge is good, is solid, we have to go back, clean the data, retrain with knowledge-based model, and come back. So the answer is, the first step, ignore what the expert said. It doesn't matter. Okay, from my personal point. Even if there are doctors, there are economists, the data know better. That's for sure. Okay.
Thank you very much.
Is there any other question? Okay. Thank you, Dr. Kypriakis.
Thank you.
For your nice workshop.
I want to thank the students, PhD students, correct or not? The team. Okay. The team from Patras. Okay. For participation.
They did their best.
I know.
Question about one plot with the box plot there. Sorry.
Yes.
I kept for myself for the last moment. Okay. Yeah. These box plots are on train or test data set?
Dummy data.
No, it's based on the accuracy on the test
or
the train test?
It's from dummy data, just for the presentation.
Okay. But actually, we are going to do it with the train or with test data?
Test.
With the test. Okay.
Nope.
So, in order to answer, 9 times 10 seconds plus the third one. First of all, because it has less variability, and second, because there is explainability. I can understand with the numbers. With them, no.
Yes. Explainability. Okay. Thank you very much.
Okay, thank you. Let's proceed. And now we'll have with us Kyriakos Giallourakis. He is the president of One Health Students in Greece, and he will be together with Christos Rafaelianos, the vice president of the same group. and Nikolaos Papadopoulos, who is president of the Institute of Electrical and Electronic Engineering and Medicine and Biology Society. And I believe we'll have these persons online. Is that true? Okay.
Hello, everyone. I would like to ask the organizing committee how the other team members participated connected in Zoom.
I'm sorry, we can't hear you very clear.
We cannot hear you very well. Can you repeat, please?
Yeah. Now, any better?
Kind of.
So and
No,
so.
kind. Okay. Okay, I'll try to speak as loudly as I can. Okay, so hello everyone, and thank you everyone for being here today. At first, I would like to thank the organizing committee and for the very honorary invitation, and especially Professor Vandarakis and his team. In any case, I would like you to please forgive my accent, just to be clear. My name, let me just make a share of my screen. Great. Can you see my screen?
Yes.
Yes.
Lovely. So,
Can you make it full screen, please?
Yep. Great. Can you see it in full screen?
No, unfortunately, not yet.
Not yet.
Now it's okay. Thank you.
Great. So, it is my pleasure to present to you the work, the vision, and the impact of One Health Students Greece, the largest interdisciplinary student federation in Greece, dedicated to promote the One Health approach. Me and my colleagues are going to present to you three topics regarding empowering youth for One Health in the digital era. One Health Students Greece was founded in 2021 and officially established as an organization in 2022. Today, One Health Students Greece consists of more than 5,000 students nationwide, representing a wide range of scientific backgrounds from universities and institutions across the country. Our federation operates in alignment with the International Student One Health Alliance, ISO, which is a member of the World Health Organization Youth Committee. Through this partnership, we aim not only to empower Greek students, but also to connect them with international networks, initiatives, and opportunities within the global One Health community. Let's talk more about our organization. Our goal is the core of our mission, to be more precise, lies in the One Health approach. It is you have talked about it in the whole conference these days, which is a holistic interdisciplinary framework that recognizes the interconnection of human health, animal health, and environmental health. Our goal is to cultivate the next generation of scientists and health professionals capable of addressing modern health challenges, zoonotic risks, and emerging threats through collaboration, innovation, and prevention. We more or less focus on raising awareness, organizing scientific events, and fostering partnerships that bridge the gap between different sectors and disciplines, from medicine and veterinary science to biology, agriculture, public health, and environmental sciences. Our organizations brings together students under a common purpose, building a safer and healthier future through One Health. So, our structure as an organization, how do we operate? This is the board member of 2024-2025. And as an executive body, there is the president, followed by the vice president, the secretary general, the treasurer, the event manager, and the social media manager, and 2 more members. But as an organization, we don't have individual members. Because, as we said earlier, we work as a federation. So our members are student scientific organizations. And they, each year or each 2 years, they elect a team representative who is later added to the advisory and executive body. So, as a federation, we are being strengthened by multiple organizational members. And so that is our goal, to have more representations from each member into our core executive body. So, our members. Our members are, until now, the Students' Association of Faculty of Public and One Health of the University of Thessaly, the Scientific Society of Hellenic Medical Students, the Greek chapter of the International Association of Agricultural Students, the Greek Pharmaceutical Students Federation, the Aristotle University of Thessaloniki's student chapter of the IEEE EMBS, the BioAuth team, the Aristotle team of Pharmacy ATP, and the 2 Greek chapters of the International Veterinary Student Association. So our activities, our achievements until now, up to date, include at first our representation at the International Student One Health Conference Push-Through of the WHO Youth Committee. And after that, our first nationwide seminar was about antimicrobial resistance. And then we have done many mini conferences, one-day conferences about various topics regarding biosafety, genetics, food safety, biotechnology, and many more. And through that, through our activities, we have collaborated with many universities. We have collaborated with more or less Greek universities across Greece, including the University of Thessaly, the Agricultural University of Thessaly, the Aristotle University of Thessaloniki, the University of Crete, and many others. In addition, we collaborate and we like to collaborate with numerous scientific institutions, national authorities, and research centers and professional associations across Greece. And with that, One Health organizations that are in Greece. These partnerships allow us to expand our activities, engage students with high impact projects, and promote the One Health mission at both national and international levels. Together as an organization with those partners, we develop educational programs, scientific events, and opportunities for students' involvement in areas such as we said earlier, biosafety, genetics, biotechnology, public health, crisis management, and food safety. One Health topics in general. Here I present to you some of our posters of our previous events about biosafety, genetics, biotechnology, food safety. And closing, One Health Students Greece is committed to shaping a new generation of scientists who understand the complexity of modern health challenges and the importance of interdisciplinary cooperation. That is our core value. Our mission is clear: to promote One Health, empower students, and foster collaborations that contribute to a healthier, safer, and more sustainable world. talking about sustainable world in the digital era, most of our events are being not only hybrid and more or less online, because our members are across Greece. We want it to be more or less sustainable. So we try to be accessible to every student, and that's why most events are being held online. I would like to thank you everyone very, very much for your attention. Thanks again for the organizing committee and Professor Vandarakis. And very happy to success the next my colleagues. This is Rafael Giannos and Nicola. And I will be very happy to answer any questions you may have. Thank you very much for your time.
Thank you, Mr. Yalourakis, and it was very nice, and it gave us hope to see that students are against this approach of One Health, because we have to do something with all these environmental and public health problems, and maybe the One Health approach is a solution to all this, and it's very nice that you are engaged in all this.
Yes, and if I allowed,
yes, of course.
I would like to say, first of all, congratulations for the initiative of the conference. And Mr. Vandarakis knows very well, because we have talked about this, the importance of One Health. It's very important to tell your participants that One Health in Greece is already here through institutions.
Yes.
We have the Faculty of Public and One Health, a department inside the university, and we have One Health Student Greece, which is the largest student organization in Greece. So practically, we have already established One Health in Greece in our country, which is very important, and thank you very much through this conference that we have the ability to point it out to more students and health professionals.
Yes, it is a very nice task. Is there any question or any comment? Would you like to be written in the group? I think you're open to new entrances. Okay, I will persuade some of these. And so we'll continue with Christos Rafael Giannos, is that correct? The vice president of the same group?
Christos Rafael Giannos.
Giannos, I'm sorry.
No problem. Okay, I would like to share it, so I can share it now, I think. 1 minute. It's everything all right?
No.
No?
Now it's better, and if you have a slide presentation, okay.
I have in
That's
full
perfect.
screen.
Perfect, okay.
Okay. Perfect. Good afternoon, everybody. It is a great honor to be here with you today at this important and delightful conference, I would like to say. And also would like to say sincerely thank to Professor Vandarakis for the invitation and for his continued support of young people in the One Health community. We're also very proud to be supporters of One Bridge conference, a meeting that truly connect science, innovation, and collaboration. Our topic for today's speech, it's the empowering youth for One Health in the digital area in proposals and plans for the future is my part. It's not only timely but essential because young people today live in the world shaped by technology, fast communications, and global challenges that we can see around us. And also that I would like to say all those link with the human, animal, and environmental health more closely than ever before. As students, and young scientists, we stand at it's at a unique point in time. Digital innovations give us the power to learn faster, connect wider, and act earlier. It is allows us to join forces across cities, countries, and scientific fields. Because, like he said, the Kyriakos Galourakis, were in various fields in the sun. So this is important for many fields in scientific fields. And most importantly, it gives us the tools to build a stronger one that is informed, collaborative, and ready to respond to the needs of the future. Today, I would like
Mrs.
to share
Rafael,
with you.
sorry for interrupting. We cannot see any change in the slides. We only see the first one.
Oh, okay, 1 minute. Now?
Yes, now we can
Now,
see the
yes,
changes.
Okay. Okay, I would like to stay here for one moment. And what I was saying, yes, today I would like to share with you proposals and plans that said before for the next steps of One Health Students Greece. So this is the first part, the seminars and webinars. I can show you with, I don't know if it's necessary, 1 minute, with this. Okay. And the first part that I would like to propose ideas is the seminars and webinars of the One Health Students Greece. So the digital innovation and workshops and etc. Seminars, etc. One of the most ambitious projects we are preparing in the organization is the nationwide hybrid conference, where different universities and institutes were going to have across Greece will host simultaneous live workshops and seminars that all connect in real time through one central online platform that we have. This way, each local team will be able to present its own works and approaches and experience in oneself, while the audience will have access not only to their local event, but the entire nationwide panel at the same time. This structure fully reflects the philosophy of leveraging digital innovation to advance oneself. So technology is not just a tool, it's become the mechanism that connects different places, cities, and scientific fields in one shared experience. So this is a solution for the technology itself. This simultaneous one-person presentations. So this is the one idea that we're already doing the hybrid, but not simultaneous in more than 1 or 2 cities. So this is a promising idea. The other idea, this is the seminar that we're talking about, the role of artificial intelligence in oneself, that the role of artificial intelligence in oneself that we can help with this project, with that idea, that we can talk about this in a separate seminar. We can talk about digital policy and government readiness and innovation and application practice. All of that we can share it either in the hybrid webinar slice workshop that I was talking about, or in a different seminar, because this topic is very crucial in our days. that is very important to do something like this in the near future. The next idea for this, it is the process automation that already do, but we have to improve it. For this, we have already have an algorithm that automating the certifications, but the automation is a core part in of the digital innovation. So using an algorithm for setting certificates allows accuracy, speed, and reduce human error. So in a conference like the last one that we did, we have 800 certifications. This is not very promising if you want to do this in manual. So the automation is very good and ensuring that they are we are going to send them the certifications quickly and correctly, and this will save us dozens of working hours and serve participants much more efficiently. So we'd like to do the algorithm even faster than is it now. The other thing is the international networking that we are already have, but we have to extend it. Building participants. In the coming years, we will work to strengthen our connections with international partners, universities, organizations, and global One Health groups that we are already do this, but we're going to extend this. Digital innovation allows us to communicate directly with scientists and leaders in the fields, extend knowledge, and build joint initiatives. The globalization of One Health is not longer a theory. It is a reality made possible through digital tools that remove geographic borders. So this is the other thing. And most importantly, using of digital innovation in One Health students Greece in the work groups inside the One Health students Greece, inside the organization. This is the most important thing that we have to do in our groups. So as we move forward, empowering our structure is key for this. To support long-term growth, we're developing new teams that will help us produce scientific work, communicate our message more efficiently, and welcome new members across Greece. For this, these teams represent the next stage of our organization. They combine teamwork with digital tools, allowing us to work faster, smarter, and with stronger scientific impact. They will help us stay connected, stay organized, and continue to grow us as a national One Health community. We are already have the social media team and the recruiting team, but we can do this more efficient with the digital innovation. So the new research writing team that we don't have it yet, but we're going to have it in the next year. The new One Health Students Greece research team will use digital tools for literature, for review collaborative writing, data analysis and scientific output. So the other thing about the scientific writing and research team is that this is easier to communicate and extend the knowledge around our scientific fields, so we can write more efficiently about this, about our reviews and our research work. Recruiting. For the recruiting, our next step is to welcome new teams that we are already told you about this from different scientific fields. With the support of digital tools, we can organize remote meetings, provide training materials, and connect members from all over Greece. For social media teams, it's obvious the use of digital in our social media teams in Facebook and Instagram. And with this, I would like to thank you. In closing, the future of One Health depends on our ability to stay connected, stay innovative, and stay united. So digital tools give us the power to learn faster, collaborate wider, and build stronger networks than ever before. So as young scientists, I would like to say that we're not just following the change of the technology and digital world, but we're shaping it. Together we can transfer ideas into action and bring One Health closer to every community. So please, if you listened with some team in the Greece, you are free to join us. This is the Facebook, the QR code for the Facebook in One Health, the Instagram QR, and in the upper side of the slide is my account in LinkedIn. So thank you all for being part of the journey, and thank you for listening. Would you like anyone to have a question or to collaborate further? I'm here to do. Thank you.
Thank you, Mr. Yannios, for your very nice presentation of the society and your persuasive words. Any question, any comment? No, but now we know where to find you, and thank you for that.
I would like to stay here.
Okay, and let's proceed with Nikolos Papadopoulos.
Yes.
Yes.
Hello, everyone. I would like to thank Kyriakos and Jesus Raphael for inviting me to accompany them to this conference. I am the president of EMBS AUS. EMBS is the engineering, medicine, biology branch of the IEEE branch of Aristotle University of Thessaloniki. Can you see the
Yes,
presentation?
that's okay.
Thank you. I would like to present you the project we worked till last year as a group. It's the iDerm application. It's a digital app for detecting skin infections. In simple terms, it's a Google Lens for skin diseases. So we found some problems that many people face when have a small rash or a skin infection. And first of all, they are afraid to visit a doctor maybe for the cost, or maybe they're just afraid of doctors. That's called medical anxiety. Or in other cases, usually in rural places, there is a lack of access to medical care. So using iDerma, those people, those patients can have a preliminary assessment for possible illnesses, and they can get and they can find they can easily find a local and reliable doctor. What are the users of the application, the customer segment? There are patients with skin diseases who can immediately find an assessment for their condition, or online doctors who can promote themselves through the app and find clients. It's important to mention that iDerma is not going to replace the doctor, but it's going to give an assessment to a patient and instantly promote a doctor that they can visit through a review system. So let's dive in the application. Opening the app, you find the sign up or the login screen where you can have your own profile, your own folder, in quotes, with any scan you have made to your skin. In your home screen, you can find the profile section, the Make a New Scan button, where you can enter the main purpose of the application, and also some doctors nearby. Pressing the Make a New Scan button, you are greeted with the instructions on how to use the application, where you can see it's not something difficult or complex to use. You take a photo with your phone, or you upload one photo you already had taken. You wait for a few seconds for the AI model to print the results, and you get a possible disease. As you can see here, there is a place for some treatments, which is a bit an outdated photo because that idea has been abandoned. Because as we said before, it's not this application is not made to replace the doctor, but to help some people get an idea of what they might have. Also, after they get the result of the disease they might have, they're prompted to visit a doctor nearby. To make this app reality, come to life, we had a small timeline we followed. We first of all researched what this app can do. What are the possible diseases one might have? We talked with many professors, many doctors who have many professors and many doctors. Then we created a small database with our database subteam, and we had every photo of many, at least not every, many photos of the possible disease with their names, which then through our model training subteam created a simple AI trained to print the output. The software development team with the app design team created the whole design and background, not exactly AI, but backend development needed for the app to work, to function. And that's how our app was created. What are the future plans we have right now? The plans we have are to talk with some clinics so we can gather even more information, so we can update the application even more, include more diseases, and make it even usable to places, to diseases that we have not seen yet. Also, as we do now, I want to present the app at medical conferences and advertise it to medical websites as much as we can. The whole timeline or history of the app starts from the December of 2022, where we had the idea, we first thought of the idea. We started to implement it on February of 2023. And after a big development stage, we had a prototype at June 2024. At September of the same year, we were able to present it to the Thessaloniki International Fair, and that was the peak of the idea. Thank you very much for your attention. Remember to invest in your skin. It is going to represent you for a very long time. Thank you.
Thank you, Mr. Papadopoulos, for your very nice application. Can we find this application somewhere?
Sadly, it's not available for everyone.
Okay.
Because it needs to go through clinic trials, and it's quite difficult.
Okay, it will work through clinics, I believe. Is that right?
Yes.
Okay, very nice. Is there any question? Any comment? Any of anything? Okay. Thank you, all three of you, for your your contribution in our conference. And let's proceed with Professor Apostolos Vantarakis.
Well, it will not take long. Just a very short closing summary of these 4 days marathon on One Health. It's a marathon because every day was until 6:30. So you've seen that. We hope that we proved that One Health is a very variable subject. We have 4 days, four different days, with zoonosis, climate change, AMR, and of course AI. I believe that we proved through all these speeches, and I will tell you some statistics on that. We proved that we need all of us to be together to fight One Health, to fight the problems with all these environmental issues that are affecting our life and will in the future affect our life. I believe that we have not understand correctly the One Health issue. not in the Europe in general, but of course in Greece we have not understand it, and in Europe and in the world. That means we have not started cooperating as we need to do it in order to deal with this very variable and complicated problem, which needs the cooperation of different specialties. So far, working with AMR mainly and zoonosis in our lab, we have seen that it's not very easy to approach that problem because it's so complicated. We are doing sewage epidemiology, for example, in the lab. And now Zoe is working with AMR in sewage epidemiology. Even if we try to approach that problem, it is so complicated because so many factors are affecting AMR that even if you want to take the solution, it's not very simple to take the solution. We're talking about medical doctors, veterinarians, we're talking about policy, people in policy, and of course, the main problem, as I always say, is the politicians. Because politicians take the decisions. One Health problem is mainly, to my opinion, is a politician problem for politicians. They need to take the decisions. The scientists in each of these subjects, in each of these different subjects, I think they have done quite an improvement in our data, and of course we're still working. The problem is when a problem in public health is so complicated, then it needs very robust politician decisions. And that's what we lack. What we can do, because we are talking about, we are in a scientific conference, what we can do, we can still work, insist in working on that, try to cooperate between us. That's a very important issue for scientists. For example, it's not a problem in medicine, it's not a problem in veterinarians or anything else that you may separate the problem. It's a problem for everyone. It's not an environmental only problem, it's a public health problem. It's not only a public health problem, it's a problem for the community. So I believe that we need, first of all, what we can do as scientists is to cooperate, trying to understand all the hidden factors of these issues. Now, I believe that we are in a better position because we have really good tools already. Another issue that is really important between scientists is what we do here. Try to cooperate between countries, to have cooperation, strong cooperations. I like that the European Union has funded this project. We have done 3 workshops in different countries, 4th one like that in this conference, and the results of all these will be shown in the next 6 months. Our project finishes in 6 months, so we have quite enough time to show all the results and, of course, to produce new results. We need to communicate these results, of course, as strongly as we can. And we have to go one level up. That means we have to go to stakeholders, to go to politicians, and try to explain them the results that we have, and of course, the results that they are in the community, in the scientific community. I'm not very optimistic on that, because we have discussed, from my position, I have discussed quite a lot of times with politicians. They don't seem to understand the problem. And also, it's another very tricky issue. A politician's life is about 4 years. So this problem, in order to have solutions, you need more time. So most of the people do not care because they will stop their politician life after maybe 4 years. We need to take very, how can I say, long solutions. And we try to be, how can I say, insisting in applying these solutions. If you remember, one of our very special speakers,
the students, what they said, we just need to follow the guidelines. So it was really nice because when a student, a high school student tell you that, what can you answer to that? You have to just follow that. We don't follow the guidelines. So that's another issue. We need to persuade to follow the guidelines. The guidelines in anything, AMR, zoonosis, whatever. So in this 4 days conference, we believe that we showed to the people 450 registrations. We had about more than 60 speakers, 16 sessions, 3 workshops, and more than 11 countries participated. So we hope that we show to everyone that the complications and the complexity of this problem. And we hope that the outcomes of our conference will stay and will try to produce some important, how can I say, like we call it, white book with some guidelines for stakeholders. It's one of our aim in the project to go to stakeholders and discuss with them about not only the results of the 3 workshops and our conference, but in general about One Health. We're planning to have next steps these 6 months. Probably I'm going to discuss with the officer about having some webinars following these. But in general, we need to understand that the complications that we have seen through all these speakers in these projects. Some of the audience, for example, told me, oh, it's too scientific, it's too research. Another one told me, for example, oh, it's like we talk in the community, because it's so complicated. It's research and the community at the same time. It's something about animal, it's about something about human health, of course. It's chemistry, it's physics, it's AI, it's biology, it's everything. That's why we need to have all the specialties on the boat so we can work together. I love this opportunity to discuss with so many different people and to have so many different scientists here. It was a little bit tiring to have 4 days a conference until 6:30, but I have the pleasure to feel now full of experiences, knowledge, and of course people that I know that they're working in some aspects of this very complicated issue, like One Health. I would like to thank everyone of the speakers that they came here or even online, they participated online, because they offered from their side a very important part of this conference. I would like to thank our team in the project. Uli, thank you. I'm talking to you because you are very, very strict, being here all the time, and your team, of course. Thank you very much. And all the team, all the groups that they are here from the project. And of course, finally, I would like to thank my team in the lab. They were very, 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 very well organized, the conference. I have to be honest with you, they organized themselves. My participation was very, very limited. So I would like to thank them about all this effort organizing this conference. Thank you, everyone. And please keep connected with the next steps, because we need all to be together, as the student said. We have a variability of speakers from 70 up to Agis Chouros, who is 72, to speakers of 12 years old. So that means it's variable, everything. So thank you very much, and I hope that I can see you in our next steps.